Eddie with us. Eddie is an experimentalist. He has been working in the field of dendritic spines and calcium dynamic, I think, for more than 20 years. He is a professor at the Weizmann Institute and I guess also somewhere in Russia or in the previous Soviet Union. And
still in Soviet Union.
I don't know, he can tell us about this. And today is going to talk about the role of calcium. So please, Eddie, we are very happy to have you here.
Thank you. Good morning, everyone. And let me first of all thank organizers. It's really a great meeting. I wish every meeting would be like this, because the atmosphere and non-formal, conversations, which is absolutely great. And I have actually two news, bad and good. The bad news is that I'm not a modelist, I'm not a mathematician. And the good news is that I understand it. So I'm sorry, it will be a more experimental talk, but I understand that you guys are interested in concepts more than all kind of biological details. So I will try to share with you some concepts, or to try to put complicated puzzle of dendritic spine all together, and to clarify at least 1% of questions that we have around the dendritic spine. And but I wanted to start with a little bit far away from this question, with calcium itself, because we mention calcium here every day about 200 times. So I wanted to start with asking a very naive and primitive question: why calcium? Like, why calcium is so important for cell signaling? I mean, we all know that, and we have seen it here, that calcium is important, for example, for cell mobility, migration, actin polymerization, regulated by calcium coming from internal stores. Then, of course, this huge machinery of protein synthesis and gene expression related to calcium. I mean, in this case, calcium is coming from outside, but it can be also coming from internal stores, activating calmodulin and blah blah blah. So I'm not going to describe. This is not focus of my talk, of course, which is, fortunately, because it's very complicated and boring stuff. But in general, also channel modulation is highly dependent on calcium signaling. I mean, for example, BK channels have been described or mentioned in this meeting, and definitely they are a good example of being dependent on voltage, but also on presence of calcium. In this case, it's a chloride channel, but it doesn't matter. I mean, almost every channel in some way can be calcium dependent. So, I mean, isn't it strange for you? I mean, to me, it is quite strange. But if we go deeper into evolution and look at such a complicated eukaryotic cell as neuron is, and compare it to a very primitive prokaryotic cell, which is a bacteria, which doesn't have nucleus, and most importantly, doesn't have endoplasmic reticulum inside. It's just, its size. This is the real size. I mean, this is not real size, because neuron can be actually bigger than 10 microns, while bacteria is about 1 micron. So bacteria is at size of one dendritic spine, let's say. This is the real scale. But if we enlarge bacteria, it looks like this. I mean, nice individual, and surprisingly, it can do most of things that eukaryotic cell can do. For example, you can find in literature many ideas that, for example, thanks to endoplasmic reticulum, eukaryotic cells are able to fold proteins. And folding proteins is happening inside ER mainly, and we describe it as something special about eukaryotic cell, which is not true, unfortunately. I mean, bacteria without ER do this excellently. I mean, and very interestingly, also bacteria hold low calcium. Not only eukaryotic cells, but also this little guy has about nanomolar calcium inside. So calcium is definitely used for these cells for external signaling, not internal, not intracellular, but external. You would ask me, but why calcium? I mean, why not sodium, for example, is a sign, or potassium? And this question has an interesting answer, because bacteria can live in all kinds of environments, in salty water like sea, for example, but also in sweet water that we drink. And of course, for example, sodium concentration drops significantly in sweet water, but interestingly, calcium concentration is more or less constant in all these environments. You will see 2 millimolar calcium in sea water, in ocean, and also in trap water that we have, in bottled water, in San Benedetto, for example. It's also 2 millimolar. And it makes calcium universal external signal, because every cell has to communicate with extracellular environment. It has to open some pores or channels, doesn't matter what, and it should count for how long the pore is open. And even bacteria has entire machinery of calcium sensitive proteins, which simply count for how long time or how much a pore is open to close it. Because if it stays open for too long time, then all the internal stuff will flow out, I mean, which is not good. So it has to open some pores, but it also has to close it. So this is very simple idea, but this is the base of all future. What happens later on? Then we come to this big cell, which is, I mean, huge. And in this case, controlling calcium is very, very difficult because of the volume. Because if you compare here surface to volume ratio, and here it will be huge difference, because I will show it later. Volume of this cell can be several thousands of cubic micrometers, while here it's just one cubic micrometer, maybe 2, but not much. So what you do if you want to stay with this 100 nanomolar calcium, which is very low, really low concentration, you implement some organelle which will, you know, make calcium compartmentalisation, because surface of the cell is not enough to reduce calcium to such levels. For example, sodium is also must be controlled, but sodium in neuron, for example, is 15 millimolar. 15 millimolar, only 10 times lower than extracellular, for example, in seawater. But for calcium, it's about 1 million times difference. It's very hard. So definitely, you know, origination of endoplasmic reticulum is not to become a complicated organelle with all these additional functions of calcium signaling. Of course, it is true, but originally it was there to secure secure cell nucleus and also cell body around to keep low calcium everywhere. And so size of this little compartment between ER network is about bacteria size. So first of all, ER has to reduce calcium. This is the only way to keep such a low calcium concentration. Now coming back to one famous and important mistake, I mean, not really mistake, but I mean, we believe more or less that ER is important for protein synthesis and folding. And if you check literature, you can find many articles entitled like this: endoplasmic reticulum as extracellular space. Extracellular space is inside the cell. Nothing against authors, they are perfect. But do we call this space extracellular? Definitely no, because main difference is sodium concentration, for example, or potassium concentration. Can we say that ER holds high sodium and low potassium as extracellular space, or high magnesium, for example? No, nothing like this. The only difference is calcium. So only calcium reaches high concentration inside ER. So nothing to do with extracellular space. That's so others say here, and it's very correct, that lumen ER proteins are accustomed to high extracellular. So it's not that they require this calcium. They are simply accustomed. Why? Why is it? Very simple. I mean, at least to me, simple. Because I mean, you need some space, membranous space, to let ribosomes to sit on, to be there and to synthesize proteins. If they sit on the membrane, they like to put proteins inside in this space, because it's very comfortable then to pack proteins to make cargos and to send somewhere else. But it's not calcium inside which helps protein folding, because once again, bacteria can do this job excellently without any, ER once again. Another interesting picture, I like it very much. It comes from a famous Eric Kandel's book, textbook. Look at it. I mean, all this is just introduction to my talk, but it's very important to mention. This is G protein coupled to a receptor. Then it breaks a PIP2 molecule to two parts. One is lipophilic, which is DAG, diacylglycerol, and the other one is IP3. Now, this is lipophilic, IP3 is not. So IP3 can flow in the cytoplasm freely. But then IP3 makes very strange thing actually. It doesn't go to activate directly protein kinase, this one. No, it can interact with proteins because IP3 receptor on this store is also protein. So nothing problematic to change conformation of this protein with IP3. So it could go directly to here and do the job. But no, we see very complicated pathway, because it goes to IP3 on the store, opens it, calcium flows out, goes to calmodulin, occupies calmodulin, then calmodulin goes to here, to protein kinase, and then activates it. So all this long pathway, instead of cut it and make a direct influence. No doubt it is possible theoretically, but practically we don't see it, because calcium once was very important as extracellular signal. So you know, evolution is very conservative, and so it keeps mechanisms which work. It's not an engineer to go back and say, moment, why I need this calcium. It works, so it is kept. This is the reason. Now I wanted to go and remind you this complicated machinery, which is mainly neuronal, but also exists in many other types of cells, which is, you know, a lot of sources of calcium influx inside, which are all kind of channels, for example, TRP channel, ligand-gated channel, voltage-gated channels. All these can insert calcium from outside, and then calcium can be pumped into the store with Cerca pump, or can be pumped out with plasma membrane, ATPase, and so on, or it can go to mitochondria. So all this action requires energy, definitely. It's not cheap for cell to make these pumpings, you know, or exchangings like in this case. And if we look at the process, I'm not going to go deep into energy questions. There were excellent lectures yesterday describing all this. I just want to say very simple stuff, that almost every calcium ion pumping out or going into store costs 1 ATP molecule approximately, yes? And if you want to increase efficiency, you connect to the pump calmodulin, and then efficiency goes up. So if we go to numbers, these are the only numbers I know as biologist, I mean. Approximate volume of small neuron is about 6,000 cubic micrometers, and 100 nanomolar of calcium is about 1 million calciums totally. So if we look at small compartment like spine is in rest, it's very low number of about 60 single ions. This is the number. And when we say calcium is increased, for example, 1 micromolar is good increase in a spine, like this, for example, this is a Führer 2 imaging, this is about 1 micromolar. This is addition of 6 single calcium ions. So 1/10 of baseline and not much change. So we should remember it every time we mention, for example, calmodulin with 4 calciums required to activate it. We should remember, I mean, that entry of calcium into specific spine is few single ions, not like abstract numbers, but just few single ions. And pumping out every calcium costs a lot. Now I wanted to describe another perhaps very ancient mechanism, which was already mentioned yesterday, which is store-operated calcium entry. So ER exists in many compartments of neuron, not only in cell body, but also in axon, for example, as you can see here, the yellow, and these are boutons, perhaps, with mitochondria and so on. And if we go to adult dendrite, remember this, we can find it also in some spines. Not all of them, as you can see, many of them are just empty, or some of them have some initial segment, but not the head part, but some of them, very big ones, have a good established calcium store compartment, and we were fortunately first to show that this structure is real calcium compartment. And if you enlarge this area, you can see this structure, which is called, this is a EM picture, and this is like a model. It is called spine apparatus. Spine apparatus can be either connected to ER, but in most cases, like you see here, it is not connected. So it is completely independent structure in the spine head or in the passage between spine head to the neck. And it sort of, you know, blocks actually the easy diffusion. So every time we consider spine, head, and neck with cable theory, we have to remember this, that most important spines have this unique structure which reduces the volume, free volume here, as you can see, almost to zero. There is very, very little space for ions to pass on the borders because it is mainly blocked. I mean, you cannot ignore this fact when you make your models. I mean, nature is very, and once again, this structure is most important for big mushroom type spines. It can be secondary for other types of spines, but it is very important for these guys. Okay? Yeah, sure.
On that point, what is the affinity of calcium ions for the ER membrane surface? In other words, the way you, the language you just used there was excluded volume, but what about it being a sticky surface?
Yeah, I think it's about 1 micromolar. It can be, I mean, if we talk about circopumps, again, efficiency of circopump highly depends on phosphorylation. Phosphorylated circopump can be more efficient. Its affinity to calcium goes up, and non-phosphorylated is about 100 nanomolars, 1 micromolar, this range. So that's why we have finally such a concentration inside spine as well. But mainly for these big spines, okay? Okay, now, yeah, sure.
Have you looked at what's on these structures? Like what kind of receptors might be there? You mentioned pumps.
Very good question. Yeah, excellent question. Thank you. We're working now with David on this stuff, but just very briefly, we do find ryanodine receptor on it. We do find a circopump. We have to understand how are they localized. Is there any specificity in this localization? I don't want to say it for the moment, but because this is the main part of our work. But definitely, I didn't see any IP3 channels on these particular structures. Oh, this is mainly cortical or hippocampal neurons. Yes, I didn't mention it, but you know, there are, for example, very spiny cerebellar neurons, like Purkinje neurons. They don't have this machinery, but very important cortical and hippocampal neurons do have. Mainly, you know, pyramidal type of neurons, and plasticity, cell plasticity is highly dependent on presence of this structure. So all kind of LTP at level of single spine is highly dependent on it's not in my presentation, but in general, if you knock out this structure, cell loses almost 100% loses its possibility, its ability to create LTP. So it's a very important structure. Okay. Now I wanted to go back to very ancient mechanism which has been described yesterday by you, which is store-operated calcium entry, and the rest of my talk will be about this mechanism, and you will understand why is it so important, at least in my view. But just briefly, I will remind you that store-operated calcium entry is built with two main proteins. One of them is ORI. There are several ORIs, 1, 2 at least, maybe also 3, and also STIM. So ORI is a pore, ORI is the channel, calcium channel. STIM, as it was mentioned yesterday, is not channel. It's just a signaling protein which has two tails, two arms. One arm is, as it is believed, inside of calcium store. It should sense calcium. The other arm has ability to be connected to ORI channel. And when this connection is established, like here, of course it's a cartoon, you understand, then calcium flows in. Very interestingly, it flows in the cytoplasm. Even though calcium store ER can be just next to it, but still calcium first flows to the intracellular space. Then perhaps you have to put some pump nearby as close as possible and pump calcium in, which is also, you know, energy required situation. So it can be a good, excellent question to say, why don't we put it directly on the plasma membrane with some calcium selective channel, and we will just keep it always open, and store will be always perfectly full, because it will have always 2 millimolar of calcium as you have outside, but it doesn't happen. So definitely it plays some important signaling role rather than replenishing the store. And I will try to show you some mechanism which can be very important only for neurons. I don't generalize, I don't try to explain everything here about other cells, because this mechanism actually is very universal. It exists in almost all cells, but finding this mechanism in neurons is very strange. I would explain you why. Because most of cells don't have calcium channels. I mean, they use calcium because they hold it at low level and have all this machinery for internal signaling. So they need some little portion of calcium which should come thanks to this mechanism. But neurons, and also muscle cells, but mainly neurons, have huge amount of, you know, calcium sources. All the channels are GLUAR subtype, NMDA, voltage gated calcium channel. All these channels are sources. So it looks like a neuron would solve the opposite problem, to reduce calcium, to avoid calcium entry, or to pump it out, not to create a mechanism for replenishing calcium. So it's hard to imagine, you know, such a situation when a neuron needs a store-operated calcium entry, because there is always extra and extra calcium. But still, if you do immunocytochemistry, you can see this, of course, in soma, but we are not focused on soma. We want to see spines and dendrites. I didn't include here axons, even though it also exists in axons. But let's focus on the dendrites for the moment. Red is ORI, this guy, and blue is STIM. In this case, it's STIM1. I will later show you why is it important. So you can see that in some spines they are colocalized. Stem has tendency to be in the dendrite, and ori has some tendency to be in spines, in some of them, not all of them. Small ones don't have it in this case, but for example, in this case there is definite colocalization of these two. Okay, this was for us first evidence that something interesting happening there. And then we started to do huge, very boring immunocytochemical work, because it's really hard and boring. And we found, first of all, mechanism works. I mean, we can take piece of dendrite and apply these two antibodies against STIM and, in this case, pay attention, STIM2, not STIM1, it's important, and ORI1. So this is just normal case when ORI is in some spines and STIM is mainly in the dendrite. But if we keep cells in calcium-free extracellular medium, we can see after a while unification very clear between, not in all spines, but in some of them, between ORI and STIM. And for STIM1 it happens less. Also, if we check ORI2, it doesn't look to undergo same rule, and it mainly stays in the on the shaft, in the dendrite. So if we want to summarize, I mean, believe me, we have thousands of these pictures. First of all, we have to say that under minus calcium condition, ORI hardly changes, just a little bit in the most important mushroom spines, before, in free calcium, and after that. ORI2 as well. STIM1, no change almost. But STIM2 has huge relocation ability. You see, it goes up in these spines, it doubles, if not more. Okay. So if we check for couples like ORI1 and STIM2, again, I mean, in mushroom spines it goes up from control condition to calcium-free condition, and reduces after calcium is brought back. For the others, we don't see much this tendency, sometimes a little bit, but nothing compared to this special pair, ORI1 and STIM2. If we check only net difference, again, I mean, this pair is most pronounced. So it was very interesting observation for us. And now I will tell you a really crucial thing. Yeah, sure.
For the relocation of ORI, do you know?
No, ORI stays there mainly, but STIM. Yeah.
STIM, excuse me. And for the relocation of STIM, is it moving on the membrane, or is it a new synthesis?
No, STIM, yeah, excellent question. I mean, up to now people would believe that STIM is inside ER, right? Mm-hmm. So one arm of STIM is inside ER, so it should flow along ER membrane. This is the only way it can appear somewhere. So we have to believe that all these spines where STIM appears should have connected ER protrusion into spine head to let STIM go there. Otherwise, no way. So it's a very good question, because you have something else to say?
No.
Okay, so because this is most crucial. I mean, really, I wouldn't believe myself if I wouldn't do this experiment. If you check ER in young cultures, these are cultured neurons, about 1 to 2 weeks in culture, young ones. You don't see endoplasmic reticulum in most of spines. I mean, most of spines don't have any ER there. Still, we have ORI and we have STEAM inside, no ER. So this is the really strange and I would say unexplainable situation, unless you assume that STEAM can stand alone. I mean, can be not related to any membrane, any endoplasmic reticulum, because there is no endoplasmic reticulum in these spines. That's all. I mean, no doubt about it. It's very easy to check, very simple experiment. No, I can show it in these pictures because like all fluorescent channels are occupied. But in general, I mean, believe me, that's true. Yes, sure.
So just about the ori in the spine, so is that more or less stationary? So it's only the stem that actually diffuses in and out. So it's not the other picture where you have both ori and stem diffusing. So essentially that ori domain is stationary over time.
Great. We are exactly on the slide to answer this question. Very good. Thank you. You are exactly following the ideas. Which is very good. Next question for us was, I mean, if ORI is stable and it stick to some place, or it can also move, and what is the role of this ORI in general? Why it appears in some spines and it doesn't appear in other spines, and so on. So we decided to make a plasmid with this ORI and to follow movements in life. And now please pay attention on these little guys which look premature spines. And again, red is ORI here. So I will play the movie and you will try to follow some location here. Yeah, sometimes it goes out of focus, of course. What can we do? I mean, this is very tiny. You see, ORI right at the edge. And for this guy, pay attention to this one, to here and to here maybe. You see, ORI is right at the edge, very edge of the spine. Also here. Sometimes it goes out of focus. Well, pinhole is very close, so but it will reappear here. Yeah, you see, right at the edge there. So it's very mobile in this case. And
what's the time?
it's approximately real time. I mean, seconds. Yeah, I didn't speed up much this movie. And very important movie comes here. Of course, you also have some ORI at the edge of these little protrusions at the head, but please follow some puncta which you will see here. For us it was real, I mean, new stuff. And please try to correlate appearance of these protrusions with, I will play it again with your permission, try to correlate appearance of these protrusions with presence of ORI puncta there. So there is a clear, more or less clear correlation between appearance of ORI puncta and creation of these protrusions. Now, next question we wanted to, one more movie, just a short one. Again, you see this spine is able to send some, this is very big enlargement. So it was a very important question for us to understand if this ORI is functional, because I mean maybe we see something artificial but it's not functional. First of all, we have checked if we can puff calcium next to spines and see some calcium transient. So this is line scan along the spine, and with initial 0 calcium, there is a small increase, but if you keep cell long enough in calcium free, you can see, I don't know, maybe we should switch off light, I mean, I don't know, but you can see very clear response in the spine and also in the dendrite. Then we went to specific spines, and it was very complicated work because we had to find same cell after the experiment to make immunocytochemistry and to find if these spines have presynaptic terminals. So blue here is the same cell after fixation, and blue represents presynaptic terminals. So please check this guy, which has green ORI and blue presynaptic terminal. Red curve is this spine which is occupied with ORI, and the other spine on the other side is not. So this is this I don't know, violet curve, so it has very little calcium transient. The same here, even though it's a long spine, looks prematured, but still it has ORI inside, and it shows very nice calcium transient following 0 calcium and calcium puffing there. Okay, so this is actually the pipette. I don't know if you can see it next to the spine, very small pipette to puff calcium. Okay, so if you summarize, you can see responses in ORI positive spines. Some of them don't show any, but most of them do show, while ORI negative ones don't. I have to speed up a little bit to be in time. So next important experiment was to use broken ORI or constitutively active CA ORI. So we transfected these cells with the guys. So this is control responses. Red is dendrite, blue is spine. This is normal. This is dominant negative. Still some responses because we have a portion of natural. We didn't knock out the natural one, but responses go down. Rescue, like co-transfection with the normal one, recovers it partly, and constitutively active gives good responses. To summarize this, just to show you what you have seen already: control, constitutively active, big responses, and dominant negative, much smaller ones. Okay, much smaller ones. Now, very important point about spines, because if we try to link ORI presence and spine appearance in this young culture, we should see some difference. And definitely, this is typical control picture with normal spines. White arrows show classical mushroom spines, and blue ones or cyan is just a phylopodium in this case. So very few philopodia in this case, but dominant negative. You see completely different morphology. Very few mushroom spines and a lot of phylopodium. Rescue a little bit compensates, not completely. Constitutively active has very interesting view, because besides normal mushroom spine, it has lot of small spines, you know, like it tries to initiate more and more and more. So if we summarize, mushroom spines are at equal concentration or number in control and constitutively active, in dominant negative much less, and short spines, these little short spines, are very high in constitutively active, while Philopodia, like dominant at a negative, is the champion for Philopodia. Okay, and in terms of presynaptic terminal, again, I mean, there is much more here and here than in dominant negative. And this is a summary of the result. I will go fast. And another check was to use siRNA, for those of you who know, to knock out the gene, you know, the mRNA for ORI. So T is targeted, and you can see it goes to this typical, you know, typical Philopodium structure, and non-targeted, which is scrambled siRNA doesn't change morphology, and some electrophysiology physiology just to show that number of events in the targeted guy goes down, while amplitude is the same. Okay, and now do we have some clear evidences for ORI and new spine appearance? So I will simply show you several movies. In this case, you will see it by yourself, some association between red dots and new spine appearance. And this is very nice example, I think, because it creates a real spine with nice head. And always I go back to same cell and check if it has also presynaptic terminal. Some other examples, just for beauty. I mean, I don't have to comment much, just enjoy. I mean, appearance of spines exactly at the location of ORI. No much comment. So first of all, you can see ORI is very mobile, at least in young cultures, and it is highly associated with this spine appearance. Maybe some other, well, I will skip it, doesn't matter. Here we have tried to stimulate, not only to see spontaneous spine appearance, but to apply glutamate. This is not movie, but you can see in this area we applied glutamate and spines appeared, and some movie for this is here. Like we uncage glutamate here, and after 2-3 uncagings, some new spine appears. Yes, please. You need microphone.
I have probably a very naive question, but it looks like the spines appear before ORI arrives.
That's a good impression. You have good eyes, by the way. It's mainly, we have checked it. It's mainly because confocal takes two images, and it doesn't take them simultaneously. First green comes, then the red. If we do it vice versa, it looks the opposite. They usually appear together, but to be clean with our imaging, we do sequential images. So this is just to summarize, and also to mention that for young cultures, 10 days in cells in culture, it happens much more than for 20 days old cultures where most of spines are already established. So this is cumulative again to show that this is just a normal random appearance, and in Ori plus there is more chance to appear, nothing special. So our initial model to think that store operated calcium entry with association with ER can create spines. But now, after seeing that actually ER is not there, we put big question here, and here, and we ask ourselves, does steam need ER to go into spines? Because for this moment we don't see any proof for this theory. So my working theory, don't catch me on this, is that maybe steam considers a spine as a compartment, not ER as a compartment to measure calcium, but spine itself, little spine with tiny neck as a compartment to sense calcium inside and to tell Ori to open and to flash calcium inside. Why? Because we are talking about premature spines which don't have yet presynaptic terminals, these little thin protrusions, and for these spines Ori can be main source for calcium to be initiated and to establish some shape. Then axon may come, arrive, and connect to it. Okay, but this is just a theory. I mean, I don't have yet proof for this. But what I can show, this is completely preliminary data, but real data. If appearance of Ori in spines may truly flash calcium, because up to now we saw only induced but not spontaneous effects. So please follow this. Once again, red is Ori, blue is some spine, green is calcium here. So you will see that Ori enters the spine at some moment, and you can see that calcium goes up in the spine and then it leaves. So if we put several cases together, we can consider that for some spines, first Ori appears, so the red is increased because of Ori, then calcium can go up, and then Ori leaves and calcium is recovered, and without Ori nothing happens, it's just a straight line. So in some cases Ori appears but no calcium flash happens, at least we couldn't record it, but in some cases, in this fraction of cases, it's co-appearance of Ori, which is here, and calcium level, which is here. So in some fraction of cases, you see it's a big amount of cases. There is some correlation between these two. This is completely preliminary, never published anywhere. It's just for you. And very last point of my talk is oh yeah, I didn't show it as well, but again, Ori and some calcium increase maybe. You will see nice protrusion, I guess, coming out from this guy. It's completely young culture, so it likes to send, like to send. I will skip this. Maybe I'll show this one, which is red Ori, and you can see that calcium is colocalized with it. Sometimes you can see very bright green dot, which is calcium at the edge. Okay, so another important experiment was to apply so-called chemical LTP, which is part of plasticity. So if we take culture and apply some excitation for a while, then usually we see new spine appearance. And this is just little addition, functional addition to what you have already seen. These guys are new spines with white arrows appeared following chemical LTP. But for, as you have to remember, dominant negative guys, which have mainly philopodia, they also create philopodia. They do create some new protrusions, but mainly philopodia, not real spines, as you can see here. This is just little addition, and this is the summary of this. So final part was related to, if you remember, we have started to speak about local stores. Is it still correct to say that for mature spines, which do have this stores spine apparatus, which is marked here in very specific locations, you see in the necks of some spines, here, very nice location. This one is very nice, I like it very much. Can we say that actually ORI is sort of colocalized with this guy? So can it still play classical role to replenish calcium in these little compartments? And the answer is perhaps yes. This is also completely preliminary, never published anywhere, but you can see this nice colocalization, not complete colocalization, but green is spine apparatus or calcium store, red is ORI. No morphology sign here, so you don't see how big the spine is, but you can see that guys are colocalized in some cases, and maybe here. Actually, this is a movie, but it doesn't matter. I will skip it. Also here, red, you cannot see it well, but this is a store and some ORI nearby. And so if we analyze this, we say store plus ORI, very big chance. ORI, puncta can be anywhere else, so low chance to be colocalized with store. But if we see some store, ORI is nearby. That's mainly the story I wanted to tell you. So once again, part of it is preliminary, and this last part is just ongoing now. So next time if we meet, I can tell you more about it, or David can tell you more about it, because we are planning to do some work on it. And regarding conclusions, you can read them. I mean, ORI is important for spine creation, new spine appearance, and it's functional, and it can bring calcium, and steam can appear in some spines without having calcium stored inside. And finally, my main collaborators, Menachem Segal, lab technician, most of work was done just by us. Thanks for your attention.
Thank you, Edu, for this very nice and didactic presentation. Let's open the session for questions.
You see a difference if you have STIM 1 or STIM 2 regulated?
Definitely, definitely. STIM 1 doesn't like to go into spines following zero calcium, but STIM 2 likes. So this pair of ORI 1 and STIM 2 likes to go to join in spines. STIM 1 or ORI2, much less.
And you tried also to play with the calcium concentration, that because it's known that STIM2 has a higher sensitivity to calcium changes.
That's right. Yeah. We are trying now to control calcium with some chelators to be very precise. I don't have yet data, but that's a very important point to go. Right. I agree with you.
I didn't quite catch how the presence of or the proximity of presynaptic terminals was is that happens after spine formation? It's not that they're seeking presynaptic terminals that are nearby, it's rather that they develop and then it's the other way around or something.
Excellent question. Yeah. I didn't stop on it, but it's a very important question. I mean, of all we have seen up to now, I can conclude that usually, even in young cultures, axons come first and they can be already on the dendrites. Then, but they are not functional yet. Then ORI may come and create this protrusion to put this piece of membrane a part of the dendrite. Perhaps it's very important, as we have discussed. So to make thin neck and a compartment which is isolated from the rest, and only after that it becomes really functional. When it becomes functional, it can get calcium store spine apparatus inside. And this is typical sequence of events with all kind of exceptions.
Yeah. But it's only at that stage that a real working synapse forms, is it?
Exactly.
Yeah.
Exactly.
And so another question, ignorant question. So what's the role of the what are the filopodia destined to do? Why are they there?
Oh,
I mean,
yeah, very good question. I mean, I ask it myself all the time. In my view, but this is just a speculation, as you can understand. Creating some calcium is so important to create something that it has to be there. It has to create a gradient. And this prematured spine or even piece of dendrite with maybe axon on it or nearby, maybe axon can send some signals like telling that I am here, so create a connection with me. But still, to create this structure, you should produce some calcium gradient. But this membrane doesn't have any AMPA channels. Even axon would release glutamate, it wouldn't affect. Maybe an NMDA is there, but an NMDA by itself is not enough to depolarize. And so, in my view, ORI at this early stage is important to produce this local gradient, and then process is actually triggered. But this is just a speculation, as you understand.
So why would you have sometimes spines where the ER is connected to the rest of the dendritic tree and sometimes not?
Oh, yeah, you guys ask very good questions. Immediately one can see you are theoreticians who used to think, unlike biologists who like to follow experiments. Once again, it's just a speculation. I think for a very mature spine, independence of calcium behavior is very important. I think many protein trafficking, for example, when spine undergoes LTP event or whatever, is local store dependent. And even I would speculate and say that STEAM and maybe some other calcium sensors can actually count these events for specific spine and say, oh, this guy works hard. It's a very important spine. Let's bring more proteins to enlarge it. And in this case, you must create a store, a local store, which is completely independent of the rest of ER. So in this case, that's actually what we see is completely independent spine apparatus, which is well developed, big enough. Capacity is not so big, but as we could see, number of ions are also just few, you know, 5, 10, I don't know, 20, not much. So in this case, it can be really independent. For younger ones, it may be still important to be connected to ER because sometimes rest of ER is the main source for calcium to go there. So this is my rough explanation.
And so, Eddie, just a final question. From an experimental point of view, what is known about ORI and STEAM after LTP protocol or after LTD protocol? Is there some evidences of changes in one direction or in another direction?
As far as I know, you know, we are the only people right now working on this, and I don't know any works related to. But as I show you, perhaps chemical LTP, for example, you could see a new spine creation is broken when you use dominant negative ORI. So it's not a direct evidence, but sort of evidence to say that ORI is important for at least morphologically. I don't know functionally and so on, but to create a real mushroom spine, not a phylopodium, you need a complete functional ORI, otherwise you will create this long, thin, meaningless philopodia and not a real spine. So this is sort of evidence.
Okay, thank you very much. Let's thank Eddie again.
Thank you.
So I think we. Okay, so we are now resuming this morning session with Fred Pincet from École Normale Supérieure in Paris. Fred is an experimentalist and is working on basically what happened in the presynaptic terminal and more specifically under the vesicles. Please, Fred, you can announce your presentations. We are very happy to have you here to show you what to show us what you are going to show us. Thanks.
Thank you, David, on Crazy. That's it. Okay, so good morning, everyone. It's not a day with good luck for you today. I'm not presenting it right, but after Eddie, it's going to be even worse. Okay, I'm not going to present any modeling, and worse than that, there's going to be no cell, no calcium. Just very simple, basic techniques. So what I'm going to try to do is to show you two techniques that we have developed to look in vitro, to make in vitro a recapitulation of synaptic transmission. So it's going to take maybe half of my talk, I don't know how long, just to present technical details, how these techniques are made. And if you have any question or an interest, or even you who is watching on internet, if anybody is watching one day, you can contact me at this address, and we'll do whatever we can to help you technically. Never know. And that's it. So I'm going to start. So what do we want to mimic? We want to work to mimic what happens during synaptic transmission. So you know the cycle of the vesicles, I'm sure, of synaptic vesicles. All of you have already seen a diagram like this. You know there are, for the fusion part, proteins that are involved. I'm going to present them more in detail. Mathieu mentioned it yesterday also, which are called snares. The whole process is regulated by some proteins, notably MONC18, synaptotagmin, complexin, and MONC13. I'll talk about synaptotagmin and complexin a little later. This process is highly regulated, the neurotransmitter release, and it's also ultra fast, which is very specific to neurons. It takes typically, it's a sub-millisecond event. So what we're going to try to recapitulate is just this part, not the recycling of the synaptic vesicles, but the docking, the priming, let's say the binding to the membrane. Then there's calcium intake that triggers fusion, and the release of the neurotransmitters. So this is exactly what we would like to recapitulate, reproduce these tapes in vitro, and be as close as possible from physiology. And we want also to have this observable by microscopy and by electrical measurements. So there are many requirements to achieve this level of observation. So I made a list. It's a little harsh, but is something wrong with no, okay. So there are several types of requirements. First, we want to mimic the membranes. There are two membranes. There's the synaptic vesicle and the presynaptic plasma membrane. The synaptic vesicle, it's really not a big problem. It's kind of easy to make small unilamellar vesicles, which I will call, I'll write it here because I'll forget afterwards, SUV, for small unilamellar vesicles. And later I'll talk about giant ones, GUVs. So small unilamellar vesicles, it's typically between, let's say, 30 and 100 nanometers in diameter. And GUVs use larger than 1 micrometer. I'll talk about it later, so I don't forget to mention it. Okay, so synaptic vesicle, mimicking synaptic vesicle is not difficult. I mean, it's not too difficult. It's easy to make small unilamellar vesicles with the right dimension. Synaptic vesicles are very small. It's typically 50 nanometers. And we can make it of any composition, lipid or protein. And we can also extract synaptic vesicles from brains, for instance, and we can work with actual synaptic vesicles. So this one we can eliminate already. It's not going to be too difficult. The problems are coming afterwards. How do we mimic presynaptic plasma membrane? We want it to be freely floating, like a membrane, meaning there's no substrate above or below, between two aqueous regions. We like to have it freely adjustable in terms of lipid composition and protein composition on either side of the membrane. Okay? So each leaflet can be adjusted independently. Okay, I already mentioned the proteins. We can insert any protein we want on either side. And we also have non-altered mobility of the proteins and the lipids, of course. So if we can do that, then we can assume we'll be able to mimic presynaptic plasma membrane. We also want to mimic the sequence of events. So there's a lot of change of medium composition on either side of the membrane, and we'd like to be able to changing this composition very quickly We also want a membrane that we can actually observe for a decent time. So if it's stable over an hour, it will help. If it lasts 10 seconds, we're going to have problems doing an experiment. So we need to be able to change the composition around it and to have a stable membrane. Then we want to observe it. If we want to see it optically, it's going to be on a microscope, of course. It's better to have it horizontal, to be as close as possible from the cover slip. We want it large enough so that it's way above the optical limits. So typically 100 square micron would be nice, or even more. If we want to do electrical measurements, we need to be able to add electrodes. That would allow us to see many events like pore opening, monitor the fusion also, size changes of the membrane or thinning of the membrane. We also want to have a high success rate. I mean, when you do experiments, you don't want to have to try 100 times before you succeed, especially if it takes a long time to try once. And we want high-speed recording. Ideally better than 1 kilohertz because we want to monitor events that are faster than 1 millisecond. So basically what I'm going to try to do is to show you we have designed a technique that allows to to fulfill all these requirements. You're going to say, okay, there are many techniques that have already been used. I've used many of them also. And they have been very useful. I'm not criticizing them. I mean, I love them. I worked with them every day. But none of them match all the requirements simultaneously. So I gave examples here. So vesicles, small unilamellar vesicles, or giant unilamellar vesicles. You'll see, for instance, you can't orient proteins most of the time easily. You cannot change the media on both sides. It's difficult to have it horizontal. I mean, I'm not going to detail everything, but you always have what is in red. It means you don't have it. You always have some requirements that are not fulfilled. So here it's supported by layer. It's not between two regions. You cannot put proteins with the right, you cannot put protein facing the substrate. Usually the mobility is altered. Here it's a bilayer black lipid membrane, so it's a painted membranes. I don't know if you know it, most of you have heard about it. This is Montal-Müller technique. So what they do in Montal-Müller technique is you have Teflon, usually, with a hole. You have a monolayer of lipid at the air-water interface, or air buffer interface. You go down, and when you go down, you have a leaflet that goes on each side of the hole, and you end up with a bilayer in the middle of the hole. It was designed maybe in early 70s, I think, so it's been in use for a while. It's very useful also, but again, it doesn't fulfill all the requirements. DIB, it's 2 droplets, so water droplets in oil. So you have a monolayer at the interface of each droplet. You bring them in contact, and you have a bilayer that forms between 2 water droplets. Okay, so again, you cannot change the medium on each side. Nanodiscs are tiny membranes that are surrounded by a scaffolding protein that protects it. So you have just a disk of membrane. So here you can change both sides, but of course you don't separate the inside and the outside. The membrane is small. Usually it's a few tens of square nanometer. And so on, and UVs, okay. So that's it. So none of these techniques that are very useful fulfill all the requirements. So I'm going to present 2 techniques. One is we call FBL for freestanding bilayer, and the other one is pore spanning membrane. You see the first one is green everywhere. So that's the ultimate one that we'd like to use as much as possible in the future. You'll understand why later. And the other one is a pore spanning membrane that we already designed a couple of years ago. And, but that doesn't fulfill all the requirements also, but I'll talk about it at the end and show you some results so that you have at least some results by the end of the talk. So first, the freestanding bilayer, FBL. So what is it? Basically, the principle is rather simple. I'll take two colors up. You have two channels. So I shouldn't. The idea is simple. We want to change quickly the environment. We thought the only way to do that is microfluidics. Okay, so we designed a microfluidic chip, very simple. One channel on top, one channel at the bottom, cross channels like this, and a tiny hole between the two. Typically, the tiny hole will be 100 micrometer in diameter. And that's where the bilayer will form. Okay? So we have two channels, and the bilayer will form in between. So how do we do that? We use, you'll see we need 3D printing technology. We use PDMS, which is a common polymer that reticulates to make chips in microfluidics. We use PDMS because, as you'll see, the squalene, that's an oil, is absorbed by PDMS. And you'll see it's very important that it absorbs the squalene. And so, as I told you, so it's two channels connected by a hole. Sorry, I'm going to move. So you'll have one channel here, and the other one like this. Okay? And the bilayer will be here, somewhere in between. All right? So I'll show you how we, sorry, I forgot one thing. And we can put electrodes on each channel and do electric measurements. And this will be on a cover slip, and we can do observations by microscopy. So how do we do this? So first, it's Paul, a postdoc of mine that's working on this. You see he's very happy next to the 3D printer. He just received it when we got this picture. So it's, I don't know if you're interested in 3D printing, but that's, how do they call it? Digital light processing, SLA, stereolithography apparatus. The way it works is you have a resin that you put inside the container here. This box, actually, it's just a video projector, like this one. It's a video projector that's facing up, and this resin reticulates a thin layer when there's light that comes. I don't know if there's UV in it. Oh, sorry, it's not what I wanted. But when the light coming from the video projector arrives, it reticulates a thin layer of resin with the shape we want. If you put just a square in your image, just a square here, for instance, it will reticulate just a square. And you have a platform that goes up and down. So it goes down, reticulates, up, and then it goes down a little less, and you reticulate the next layer, and so on, and you build whatever you want by changing the picture you do at each slice. Okay, so that's how we make our mold for the nanofluidic chip. So what type of mold do we have? We have two molds. One for the bottom channel, which is this one, and one for the top channel. So of course the mold is a negative of what we want to have. So this is the bottom channel. You see there's a, how do you call it? A thin protrusion, whatever, inside the on the mold, on the drawing. You have it will be the bottom channel. Same here, and you see the tiny black dot here. It's the small pillar that will make the connection between the two channels. So after you enter this drawing in the software of the printer, you get this. So here you have two top molds, two bottom molds. This is where the bottom channel will be. This is where the top channel will be. And in the center, you see the tiny dot That's a pillar that will connect the two channels. We put them together, so the top on top of the bottom one. That's a zoom in here, and you see the pillar, so the zoom in is a slice in this direction. You see the pillar touches the bottom channel. Then you pour PDMS, cure it, reticulate it, and it will go around this, so you'll have a whole channel here, a whole channel there, and between the two channels, you have this tiny hole that is typically 100 micrometer in diameter. You can adjust it, of course. Once you have peeled out your chip, you put it on a cover slip, and you connect tubes, and you connect them to pumps. And then you can inject. So here, what is injected here? I think it's the oil that's injected first. Yeah. So you'll see two things in this movie. This is a bottom channel. Here is a bottom channel. Here is a top channel. So this way is a top channel. Here is a bottom channel. First, you'll see oil being injected, initially at the bottom, then on top. And then you'll see buffer coming afterwards. The oil is red, the buffer is transparent. So you'll see it's directly, the microscope objective is under, of course this is just from the top. So the oil is coming in here, and then here you see the buffer is arriving, here is a buffer. Okay, it's out. So at the bottom you have buffer, same on top. Well, this is a laser light, you'll see afterwards. Now buffer has passed on both sides. But what happens is that you have squalene that is trapped in the tiny hole between the two channels. And you'll see it here. So this is the step where, that's under the microscope, we see it by microscopy this time. And here you have oil, the buffer is at fluorescent again. That's not good. That's not what you're supposed to see. Okay, what you're supposed to see. Okay, what happens is the buffer goes through here. This becomes dark. The buffer goes through here, becomes dark, but the center remains fluorescent, showing that we have a droplet of squalene that's trapped in the middle. And hopefully we can see it here. So this is a 3D view of the droplet of squalene. Yeah, so and you rotate around it and you see it's more or less symmetrical. It's really inside the hole. Okay, so it's a 3D view. Then that's when the absorption of squalene by PDMS is very important. What happens is this droplet is being absorbed by the PDMS on the side, because this is a zoom in on the droplet. So here and here it's in contact with PDMS. PDMS absorbs it, and slowly it decreases in thickness until it becomes very, very thin. So if you measure the capacitance during that time, this is typically, it takes about an hour to absorb the whole droplet. If you take picture at this level, it's very thick, and after 20 minutes it's thinner, and then after an hour, it's almost in contact, the two sides. Then if we continue and look at it optically, here is what's going to happen. So this movie it's very accelerated. Overall, it's typically an hour and a half or 2 hours. Very good. Come on. Okay, we have a problem. Okay. Okay, that's okay. Never mind. No, that's okay. I'll manage. Don't worry, thank you. So, I think I have pictures. Okay, that's pictures from the movie. What you see is initially it's uniform. Oh, sorry, I'm not doing good. It's uniformly gray, and actually this is just at this point, right before it becomes very thin, and you already see something is appearing here. And then there's a jump, and all of a sudden there's a circle, a disk that appears like this, and that spreads. And actually this disk is a bilayer. I'll tell you why there's a bilayer afterwards, but this is a bilayer. Okay? And it increases in size until it reaches the whole dimension of the hole. And typically it's stable like this for an hour, 2 hours, and we can work with it. We can, if you don't like disks, we can do any shape you want with a 3D printer. If you want a triangle, I think I saw this movie was working, so hopefully you'll see. Yeah, it's working. So here up, you see it appeared and it took the shape of a triangle. If you want to do a rectangle, you can. See, it's thinning, and all of a sudden, boom, and then at the end, you have a rectangular bilayer. It's just for fun, it's useless, but okay. So, okay, now I can't get out of it. Okay, the question is, how do we get the lipids there? Because if you want a bilayer, it's fine. We had squalene initially, but if the squalene is absorbed at the end, you just have a hole. We need to have, oh, sorry, I'm not doing good. We need to have 2 monolayer on each side, 1 monolayer on each side, and when it is absorbed, we'll have a bilayer that forms, okay, when it's in contact. How do we get the lipids there? There are 2 ways. First way is to put lipids directly in squalene. You can dissolve it, it takes a while, you have to heat it up at 37 or 30 degrees, shake it for a couple of hours, even more, overnight, and it ends up in the squalene, and then spontaneously will go at the buffer squalene interface. Then you'll have a monolayer here, and when the squalene is absorbed, you will have no more. You'll have just a bilayer. Sorry, no more squalene and bilayer. Okay, this is fine, but you cannot make asymmetric bilayer here. Okay, because you have the lipid is coming from the same oil on both sides. The alternate solution is to start with pure squalene. You flow in SUVs with proteins, if you want, and they will spontaneously fuse, because the buffer oil interface is not favorable, they will spontaneously fuse at the interface. So you can put one type of SUV on one side, another type on the other side, and you'll end up with a monolayer on each side with different composition. You just have to go fast enough so that this is densely coated. If it's not packed enough, the lipids, when the 2 monolayer go in contact, it will break, because you don't have a stable bilayer. Okay, so we have an hour. I mean, typically the waiting time before the two sides meet is an hour. So usually it's easy to do. I'm going the wrong way. Okay. So here is an example. We flew in on top SUVs that have a red dye, which is called rhodamine. At the bottom, SUVs that have green tag, but it's blue, NBD. NBD is quenched by dithionite. Okay, so we've injected it on both sides. So that's when the SUV are there. You wash them out, and then you see you have a layer that's red and a layer that's blue that are colocalized. Okay. If you flow in dithionite on top, nothing happened. It's exactly the same picture, which is normal because the red dye is on top, not the green dye. I should say blue is at the bottom. If you flow in dithionite at the bottom, everything that's blue has disappeared, meaning we really have an asymmetric system. Okay, we can do it with proteins. So here, I didn't say it, but it was T snares for fusion. So we have proteo-SUVs, so SUVs that have proteins. We flow them in only on top. At the bottom there's a monolayer also, of course. It will spread here. We rinse them off. Then they're not fluorescent. And then we flow in an antibody that is fluorescent, and that recognizes this side of the protein, okay, the cytosolic part. If you flow it in at the bottom, you look by fluorescence, there's nothing. If you flow it on top, it becomes fluorescent, meaning all the proteins are with the right orientation. And we can put a secondary antibody, it works also, of course. So basically what this says is that we can make an asymmetric bilayer. We can put a protein, any transmembrane protein that is in an SUV, and put it with the right orientation. Okay, if we wanted to have one that's facing the other way, we would have flown it in at the bottom during the formation of the bilayer. Then we can check the mobility of the protein. So I'm not sure this movie will work, but yeah. Okay, apparently it is. So we don't see much. That's a fluorescent recovery after photobleaching. I'll start again, maybe. Can I do that? Okay, I stopped. I don't know if you see on the screen. I see, but okay. So initially it's uniform. What we're going to do is bleach a disc in the center and see that it recovers. The disc will appear very big compared to this because it's zoomed in, meaning the scale changes during the photobleaching. So I started. I'll try to stop at the right time. Up. I don't know if you see, but it's bleached here. It's hard to see for you. I mean, here you see it. I don't know if now you're too far. Okay, so it's no, that's okay. Whoever wants to come can see it here, but it's not. So it's slightly it's darker here, and when you continue, it recovers very quickly. Of course, there's no reason for it not to recover. There's nothing on each side. So we've done it with lipid, NBD lipid. We've done it with the Schnoerrs. Sorry, it's square micron per second, not micron per second square. And that's a log scale. And you see when we put a peripheral protein, which is alpha-synuclein, that tends to aggregate in Alzheimer's disease. I don't know what this is. Is it me?
Do you have your mobile phone in your pocket?
I do. So it's me. I'll put it on the back. We'll see for now. What happens and what's funny is that when you put alpha-synuclein, okay, it aggregates, so it stops moving. It doesn't move, but when you put it, when you look at the lipid and alpha-synuclein has aggregated, the lipid is also becomes immobile on both sides, both leaflets, even though it's a peripheral protein. So we don't know how it works. We just observed that because we were testing different types of proteins. I wanted to talk about the stability. So we've done a lot of types of lipids. So TS is the time it takes before the squalene is absorbed. For some reason, when we have only DOPC, it goes a lot faster. We have no idea why. It's probably the DOPC DOPC. I don't know if you're familiar with it, but it's a standard neutral lipid with two chains that have unsaturations. And the TW is a workable time, meaning how long we can work with a bilayer. And if it's blue, it's good, basically. And you see for most compositions we've used, it's good. And especially for this composition, which is an asymmetric bilayer, where we can put a top monolayer that's mimicking the cytosolic leaflet of the plasma membrane. This is a good model of the cytosolic leaflet. And on the other side, we can put what would mimic the one on the extracellular leaflet. And you see the work. We can work on this more than 2 hours, so we can really do experiments. What I didn't say, we can also observe, of course, single vesicles on the membrane. So here are single vesicles. we can monitor capacitance of fusion events. This is current measurements, meaning we put a channel, well, here it's hemolysin, so it's a protein that goes on the bilayer and spontaneously forms heptamers and forms a channel. So if you put a tension between the two sides, you can see currents that jumps in currents when you open a channel. We can also measure the conductance, which will tell us how the pore radius varies in time. What else do I have here? Okay, this one is moving by itself. So that's, sorry, that's, I don't know what I did. That's single vesicles that are moving. And what else? Yeah, and this is of course compatible with fusion. This is bulk, not bulk fusion, but not single. We don't look at single vesicle fusion here. We put a lot. I'm too slow, so I'm going to go faster. We put a lot of vesicles initially. That so a T-snare, we put T-snare like before that spontaneously go on the squalene. Then we put V-snare. I didn't explain what it is, but it's Matthew mentioned it yesterday, which is the cognate snare that induces fusion in a fluorescent liposome, and what we see is that the membrane becomes fluorescent, and of course we have controls. So with this we show fusion really occurs on our membrane. So it seems perfect, but we're not there yet. This one I keep for. Since I was in fusion, we didn't look at single vesicle fusion yet. We're barely doing the fusion with a lot of vesicles. So it should be simple to move on to single vesicle fusion. Then we'd like to go to single molecule fluorescence to follow each snare, maybe by fret, when they arrive, when they bind. And the final goal, that's a holy grail, is to be able to trigger fusion with calcium. But this, it means we have to trigger, to know, ideally to place calcium, to have calcium arrive at a specific place under one vesicle whenever we want at a controlled time, very quickly. Very difficult to do. I think we worked a lot, we talked a lot with people at Leica, and I think we found a way of doing it by using caged calcium. But it's very expensive, so we'll see if we manage to get the funds one day. The idea is by turf to shine just a light in a very narrow space with a UV light and trigger calcium just there very quickly. Hopefully we can one day. We'll see. So that's it for this technique. So it matches all the requirements that I said at the start. We're almost there, and I hope in 6 months or 1 year we'll have something where we can actually do single vesicle fusion, maybe not with the calcium, but at least not with the calcium with that resolution. We can always flow in calcium, of course. But with that resolution, maybe it's going to be a little longer. Then I'll move on to the other one. So pore spanning membranes, that was done mainly by Satish. So Satish was with me in Paris 2 years ago. Now he moves at Yale. He moved at Yale with Jim Rothman. So I didn't mention it, but of course a lot of this work is in collaboration with Jim Rothman at Yale. So I go there very often, so we still work together all the time. So the idea of the pore spanning membrane is to have a membrane that's held between two on a hole that's in a substrate, let's say. So you have a hole, a substrate, and you have the membrane that's there. And here you have a membrane that's free between two aqueous medium. And you can look at single vesicle fusion. The way we do it, I'll go quickly, is we use commercial chips. Actually, I should say we're not the first ones to do this. The first one, I believe, did it for fusion is Claudia Steinem with at Göttingen. But she was working on tiny holes that were 500 nanometers. So the problem was that this is so small and optically that maybe she went to 1 micron, but the vesicle tended to aggregate next to the substrate, so it made things a little more complicated. We're working with a grid that looks like this. that has a lot of holes. We treat the surface. It's a whole process, but we published all this, so it's written in the protocol here. And you have a lot of holes in the grid, so the dark spots are holes that have 5 micrometers in diameter. We're far from the 100 micrometers that I had in the previous case, of course. And when you put giant unilamellar vesicles that have proteins, or not actually, of course, but if you want to study with proteins, you make them with proteins. So to make it with proteins, we also designed a protocol that works well for snares and for several other proteins. But there are other protocols, of course, to make proteo-GUVs, but for these ones, for snares, that's the one that works the best in our hand. And you spread GUVs on this grid. And you see, you cover the holes with the membrane. And at the end, you have a pore spanning membranes, and I'll just give you 2 results that we got with this technique on fusion, of course. I'll show you first that snares and synaptotagmin 1, are sufficient. We don't need anything else to promote calcium evoked release. And the second one is that synaptotagmin 1 and complexin have a synergistic role to prevent spontaneous fusion. Yes?
Actually, I didn't follow what was the advantage of the first technique compared to this PSM.
Okay, I should go back to the first table, but there's a lot of things you cannot do with this. Okay. You cannot make it asymmetric. It's more complicated. Make asymmetric GUVs, it's not perfect, and you never know how it will end up here on the membrane. It could go either way. You cannot change the bottom medium. It's trapped. Okay, there's a well, and it's trapped. Okay, you cannot put electrodes. I mean, I think I went through everything, but there may be a couple of other limitations. Okay, so it's not, you don't fulfill all the criteria that I had at the beginning. Okay, I could go back to the table, but I hope it's enough to convince you. So Mathieu showed this slide. I'm not going to show it. I'm going to show it again. So you have a vesicle with V snare, membrane with T snare, and as they associate, they pull the membranes together and they end up with fusion. Okay. And synaptotagmin, since I'm going to talk about it, that's a calcium sensor. It has 2 domains that recognize calcium. Initially, it's bound to the membrane probably only, but maybe it's more complicated, by the what's called the polylysin patch, because the plasma membrane is negatively charged. So the polylysin patch is positively charged. It's just electrostatic interaction. There are loops here that are hydrophobic and get inserted when calcium is bound to the C2 domains. So the green parts are, and the yellow parts, which I'm no, I'm wrong. The blue dots are calcium. Okay, so that's where calcium is binding, and these purple domains will go inside the membrane because they will be exposed when the calcium is binding. So the protein will reorient, which will release the vesicle that was held in this configuration. Okay, I didn't explain it well, but I should say it again. You have a vesicle, snare pins start assembling, but synaptotagmin goes in the way and prevents it from assembling more. So there is no fusion. It's just held in a situation like this, where the vesicle is very close, but not close enough for fusion. And when there's this conformation change of synaptotagmin, it releases the snares, and the vesicles, boom, they fuse. That's why we reach the millisecond, because it's prepared very close to the membrane so that it can fuse very quickly as soon as calcium arrives. So calcium here, when it arrives, the loops insert and the vesicles can fuse. So what we do is, here we have one more thing. We didn't put microfluidic flow chamber on top yet. We will, but we didn't yet. I see it because here we're just pipetting on top. So the way we do it for now is we inject, so we have our bilayer on top of the holes. We inject the vesicles. We wash. Then we can put calcium. And if there's fusion, we'll see lipid mixing because vesicles are fluorescent. What does it look like? I hope this will work. So these are vesicles that have, on average, 12 vamp, which is the V-snare, and 12 synaptotagmin. Before we put the calcium, so in this, I should have put it in this region, this is the type of movie we see. Okay, please work, yeah. And the red dots, of course, are vesicles. Almost. I almost succeeded. I'll try not to mess up. The red dots are vesicles, and you see they tend to accumulate. Okay, so they are clamped. They don't fuse. I mean, maybe some of them fuse, but we actually cannot see them because there are too many around them. Then you rinse. You put calcium, pipette. So no control really on the T0, so no way we can measure a millisecond resolution, have a millisecond resolution. So this is more or less the same image. And what happens? Calcium will arrive, and what you'll see, everything will disappear when calcium arrives. And then you have a clean membrane. You triggered fusion with calcium. Okay? So this just shows that if you have vamp and synaptotagmin, you can recapitulate the clamping and calcium-induced fusion. It's not everything, the whole story, of course, but what we observed is that when we put an excess of synaptotagmin, or at least when it's stoichiometric, meaning one V-snare for one synaptotagmin, we have the clamp. If we have an excess of V-snare, we don't have the clamp anymore. We have spontaneous fusion. I'll get back to it right after. But if we have less than 6, meaning if we have 6 or 6 V-snare, 6 synaptotagmins, or 3 V-snare, 6 synaptotagmins, we'll see also spontaneous fusion. What does it mean? So there's this model that came out in the Rothman lab that synaptotagmin can oligomerize and make a ring. Right now the model says we can have typically 20 synaptotagmin, maybe 18 or 24, something like that. And there would be 1 snare every 3 synaptotagmin. From these experiments, it's not fully compatible with the model. It's compatible in the sense that if we have less than 6 synaptotagmin, it goes to spontaneous fusion. So it needs to have a collective effect. It needs to oligomerize probably or something like that. As a ring, we don't know, we don't see it. But it works when there are, I said 6, when there are 12, it works. We don't know between 12 and 6, we didn't do experiments. So when there are 12, it's enough, and the 1-1 stoichiometry is enough also, which is more snares than you have here, and less synaptotagmin. So does the ring model work? I don't know. It has to be drawn and geometrically checked. Yeah, go ahead. I'm almost at the end.
Who wants to talk?
Ben.
Thanks. Yeah, that's very interesting. But perhaps you said it and I missed it. What about if you have more synaptotagmin than vamp? What?
Yeah, so it works. If we put more synaptotagmin, yeah. If we do one vamp for 2 synaptotagmins, it works also. But I don't know. I'm not sure what the threshold is. I cannot. Yeah. I don't remember exactly all the conditions we tested. We are in the process of doing it. We didn't fill up the table completely yet.
But then, roughly speaking, I mean, I realize these are preliminary findings.
Yes.
Roughly speaking, less than 1-to-1, it doesn't clamp.
Yes.
Yeah.
Less than 1-to-1. That's what we have for now. And less than 6 for sure. It doesn't clamp either. Even if you have less vamp.
Right.
That's what we have now. So maybe it's going to be less than 9 later, or less than, maybe 12 is the limit, I don't know. And the second thing we did is we didn't look at calcium anymore, we looked at the spontaneous fusion. And a calcium independent fusion. And what we did is we did experiments with synaptotagmin, Schnoerr, and a small protein called complexin, which is more or less soluble. I said more or less because it can be lipid bound. And I don't have a drawing, so okay, I'll get to it afterwards. So it's a protein that's soluble. And we'll do experiments in various conditions with various quantities of synaptotagmin or complexin or Schnoerr. So this is typically a movie that we see with, what am I doing? Okay, it's playing. Twice as many VAMP as synaptotagmin. That's kind of the case. And you see a vesicle that's here, and it fuses. Okay? It wasn't very fast, but it fused. Okay, if you look at the fluorescence, that's a disk that just shows the position at that's just measured at the position of the fluorescence that's measured at the position of the vesicle. When it fuses, it disappears. And if you measure outside, you have a burst of fluorescence, and it's a little higher at the end because the fluorescent lipid has spread everywhere. The burst is because it dequenches. So here is a brief summary of what we have observed. If we put, so what I forgot to say is, physiologically, you have in the synaptic vesicles typically 60 VAMP and 20 synaptotagmin. Okay, so you have an excess of VAMP. This was measured in a very nice paper. If you want to see a paper with a lot of data, you look at it. It's by Reinhard Jahn and others. It's called The Molecular Anatomy of Trafficking Organelle, I think. It was published in Cell a few years ago, and they looked at every single protein in the synaptic vesicle, every single lipid, everything. It's wonderful work. And so 60 VAMP is exactly what we have in physiology. We put 22 synaptotagmin, okay, it's maybe 22, which is exactly what we have in physiology. If you put 60 VAMP alone, there's no clamp, meaning the vesicles just go to fusion. If you put complexin, which is a protein I mentioned before, in solution, 2 micromolar, same. If you put 22 synaptotagmin, like in physiology, same also, it fuses. I'll go into the detail right after. If you put everything together, meaning 60 VAMPs, 22 synaptotagmin, and 2 micromolar of complexin, then there's a stable clamp. And I'm not going to talk about the last one, which is a mutant of synaptotagmin. So what happens? If you look at the survival function, meaning how long does the vesicle remain clamp, or what fraction of the vesicle remains clamp after a certain time. If you look at where there's everything, it's 100%, meaning it's clamped, it stays there forever until it's bleached, basically. Okay? If you look VAMP with VAMP alone, 60 VAMP, you see it goes down very quickly. There's spontaneous fusion very quickly. If you put complexin, same thing. VAMP and complexin, same thing. If you put VAMP and synaptotagmin alone, there is spontaneous fusion, but it's delayed compared to the case where it's VAMP2 or VAMP2 with complexin. Okay, I'm not going to show this because it's the same as before. So our interpretation is that, okay, we have 20 synaptotagmin, 60 VAMP. Too many VAMPs to be blocked by synaptotagmin. Okay? So let's assume we have a 1-1 stoichiometry. We don't know if it's true, it may be 1-2, I don't know. We have a 1-1 stoichiometry, so 20 VAMPs or 22 VAMPs are blocked by synaptotagmin. Okay, but there are still 38 that remain. Except these 38 are located outside the contact area. Okay? So they can find a T-snare that's in the membrane, but it's a little far. So it will take longer for it to fuse, to find the T-snare and to fuse. This would explain the delay here. Okay? Then this delay can also be used by complexin, because complexin binds at least transiently to T-snare and blocks basically the, it's complicated, but basically it blocks the zipping of the snares. And if it's there, if it has time to come, before the V-snare arrives, it can block a V-snare, T-snare complex that is forming and prevent fusion. If you don't have the delay, then VAMP will fuse too fast before complexin arrives. So what our model, which we're still checking, I don't guarantee it's true, is that synaptotagmin will block the snares that are located here. The remaining ones will take longer to find the T-snare, and during that time, complexin can come and block the formation of the snare complex. Okay? Which doesn't happen if there's no synaptotagmin, because if there's no synaptotagmin, the first VAMP will arrive, the first complex will form too fast. Okay? I don't know if it's clear, but that's the model we have. And that's the two results I wanted to present, and that's where I'm going to stop. Thank you for your attention. Okay, and now, David, if I can still talk, you want to start the questions?
No, go ahead if you want to.
You want to start the questions? I start the questions.
Go ahead.
Okay, so now the system looks nice, the first one, or the second one also, but since you're all here with different views, what would be good questions to address with this system? I know what we're going to address, but I think it can be addressed to many other types of processes that occur at membranes. If you have ideas, that we could try, we're happy to interact or collaborate with you or whatever. So if any of you have a good question, I don't guarantee we can do everything, but that's it. So I have the first question. Yeah.
Okay.
Actually, I was thinking about several specific questions, because I used to work with people in endocytosis, and there are many questions for the stoichiometry in endocytosis, and especially, for example, so the main pathway is clathrin-mediated endocytosis to enter a cell, and there is a big question for the stoichiometry between the two main components, which is the dynamine and clathrin. And so, this is like a type of specific question that should be addressed with this type of to see the kinetics, what is blocked, what is enhanced, etc.
Okay, that's interesting. Okay.
So, but we might discuss later.
Yeah, we can discuss it. Well, okay, yeah, we stay in touch then. Okay. Yeah, I'm interested in that. You'll be at Pasteur anyway. We can interact when you're at Pasteur. Yeah, good. Sorry.
So in this picture, Fred,
which picture? The last one?
The idea of the complexin dealing with the 40 other guys.
Yes.
Right? So I guess two questions about that picture. Firstly, you might have thought that the 40 other VAMP would not be a threat to fusion because of their being too far.
That's what I'm saying. Yeah.
Yeah, but if they're not a threat, why do you need complexin to quell them?
I was probably not. They are a threat. They can fuse. Maybe we're not 100% sure it's true. We think we have a preliminary experiment it seems to indicate it's true, but I'm not 100% sure yet, so I cannot guarantee it. But if it's the case, I think what happens is they can still bind. The tips are close enough. You can have a fluctuation that bring it really close enough.
Right.
Okay, but it will take longer, and complexin comes in and out of the T-snare, and it will probably take longer also to zipper because there's resistance. Okay, so complexin can bind, and then or you know it. So complexing can do trans interactions between two snare complexes.
Right.
So probably it's possible that it's what it does. So it takes time to assemble all this, and basically the fusion from here is longer because it's further, and it's more complex because this is a kind of rigid washer or whatever.
Right.
So you have to rotate the vesicle or something. I don't know how it works. So it gives time to create a dead-end machinery. It's not clamped anymore. It's dead-end, where some snare complex start forming but are just blocked for good.
Do you think that, I mean, is it possible then that complexin plays no role for the other 20, for the important 20 at the bottom?
Okay.
Or is that's too radical? That would be a departure.
It's possible, except there's a structure from Brunger that came out a few months ago, but a few months ago, and he said that in the binding between synaptotagmin and the snare pin, there may be complexin at some regions. It may play a part differently. I mean, it may have different parts.
Right.
So it may have one with synaptotagmin and one alone for the snares that are outside. It's just a model. I don't claim it's true. I don't know.
So I wanted to ask you two questions. The first one is, what do we know about how syntaxin is actually finding the snare that are actually at the right position? Are they just soluble protein? I mean, I'm trying to get a sort of mental picture. They're just floating on the membrane and go to the right place? How do they know that they have to go to this specific one located underneath the vesicle to prevent fusion?
Oh, you mean synaptotagmin?
Synaptotagmin.
Okay,
yeah.
I thought you said my mistake. Okay, synaptotagmin is a membrane vesicle protein. It's already on the vesicle. Okay, so it binds directly to the membrane through electrostatic interaction, and it interacts with the snares. I mean, everything is at very high local concentration here. Everything assembles quickly.
And so now, is there a possibility to add in this construction, I mean, the classical type of calcium channels to understand how, I mean, is this something feasible, or this is...
That's something I wanted to do. I wrote it in a grant and the reviewer killed me. They said, well, there are groups that are just working on this. I thought, well, you can put any protein there. I'll just extract channels or whatever, purify them and put them. The reviewer said, impossible. People have been working on this. There are groups that just are experts in this, and I trust them. I think they're right, these reviewers. They should have killed me. They're right. It's very complicated to do. You're not going to be able to solve what people didn't solve in 15 years by yourself. I mean, maybe we can collaborate with someone like that, and but it will take time. That's what I wanted to do initially. I thought, well, it's easy. We're just going to put calcium channel, and the calcium will just depolarize the membrane, and then the calcium will just go through. But no. That's why we ended up with a cage calcium solution.
Yeah, maybe one suggestion. Maybe you can go with ionomycin or something like this, or cramazidine, that you just induce pores. You know, that you have just. Pore infusion.
And then the calcium just goes through.
Yeah, that you have a more localized calcium signal. That you can maybe do.
Yeah, everything.
But for calcium channels, I would completely agree. I mean, they have 24 transmembrane domains. That's hard to keep them.
I agree.
In order.
After the reviews, I thought, maybe they're right. And I looked, yeah, they're right.
No, one question for the syntaxin. How did you play also with syntaxin? How it organized in your...
No, okay. What we did is, that's something we want to do, but it's a lot of things to do. Syntaxin is always pre-bound to SAMP25, which is probably not the case physiologically in this system, in what we did. And we didn't play on its localization. We don't know where it is. We have PIP2 in the membrane, but we don't know where it is exactly. We're just looking at the vesicles and we see them clamp. And we will, of course, but a lot of work to do.
About the localized calcium release, you said in passing that essentially you can target calcium very precisely, but it would be very expensive.
Yeah.
How exactly does that work, and what kind of resolution can you get? Like, how precise is this?
I think we can go to, it's going to be micron scale in X and Y.
Okay.
But we can isolate one vesicle in this area. We have a disk of 100 micron in diameter.
Okay.
So we can, if we have 10 spots, 10 vesicles, we can isolate them one by one. And the thickness is not going to be too much because we can do it in turf.
Okay. Oh, one more.
I think recent studies demonstrate that another isoform of synaptotagmin plays a key role.
Yes.
Were you thinking to maybe consider, say, synaptotagmin 7, which is involved in asynchronous release, and maybe investigate asynchronous release?
Absolutely. That's among the things we plan to study also. Right now we start with a minimal system and we'll expand slowly. We'll put also Monk 18, Monk 13 and the others. And slowly, I mean, it's the first experiment, so that's what we have. But you're right. Thank you.
Okay, last one.
Yeah. And just from an experimental point of view, can you combine this system with like fluorescence microscopy, like TIRF fluorescence microscopy?
Yeah, turf, yeah, we can put turf. Not on the last one. On the first one, on the new one, yes. We match the refractive index at the bottom. We've done it on just test experiments, but it works. I mean, it shifts. There are optical issues, but we solved all of them.
Okay, so let's thanks Fred again. So now we have the final talk of this morning, and then the afternoon is free, and we'll have to compensate for this free afternoon. Tomorrow we'll resume at 2 o'clock instead of 3. So we have 1 hour lunch, so we'll have to stop on time on Thursday morning. Right? So the next speaker is David. Thanks. And we are going to shift from experimental neuroscience and biophysics to parameter estimation, I guess. Okay, David. So we have, yeah.
Thanks. Thanks for the introduction. Thanks a lot to the organizers for this nice workshop and for inviting me. I'm very happy to be here. And yeah, as David already said, and as you can maybe tell from the title of my talk, this talk will be very different to the majority of the other talks so far. And to stay in the spirit with the other talks today so far, I want to apologize in advance. Though, yeah, kind of in the opposite direction to the other two speakers today, right? While Eduard Korkotian apologized for giving a purely experimental talk with no modeling, and Frederic just said he would make things even worse by not even having cells in his talk. I think I can make things even worse because my talk has no calcium in it and no experimental data whatsoever, and really not much biology. But I still hope that the talk will be interesting to the audience, since, as we have seen in the talks in the last 2 days, many of you use dynamical models to describe calcium signaling pathways, and many of those models are of spatial or stochastic nature. And many of the speakers have pointed out that those models can be very difficult to analyze. For example, Rüdiger Thuel yesterday mentioned that he would like to extend his deterministic PDE description to a stochastic setting, but he also pointed out that that is highly non-trivial in his case. And this is exactly why I wanted to give this talk, to explore if the models are developed, maybe have the potential to be useful for some of the problems that people in the audience are interested in. And in the work that I'm going to present today, me and my co-workers addressed some quite generic problems of stochastic process modeling in a, I think one can say, rather unconventional way, because rather than trying to solve them directly, we tried to map those processes and problems onto seemingly unrelated classes of models in different fields, specifically statistics and machine learning, and then try to approximate those alternative models. But let me start by motivating this work. So, as I already said, the overall topic are stochastic processes. And I will in a moment make clear mathematically what kind of stochastic process I'm talking about. But for now, let's just use a very rough description and assume we have some stochastically interacting particles or agents that undergo some stochastic interactions. In this rough picture, stochastic processes can be used to describe many systems in nature. Yeah? The one I'm most interested in is systems biology. And actually, here I've been lying. I said there would be no experiments, but I have some experimental data, not my own, unfortunately. So this experiment was done by Eduard and co-workers in 2002. And what they did is they took E. coli bacteria and tagged two different genes with two different colors, yellow and green. And they took two genes with basically identical promoters, which means both genes basically experience the same influence of transcription factors, polymerase, and so on. So one might then assume that the genes become expressed as at constant ratios all the time, because they have equivalent promoters. But as you can see here, the colors fluctuate strongly in this population. So one might think, okay, well, maybe they are just not equivalent, yeah? They just do differ from cell to cell or something. But as it turns out, further analysis showed that the number of proteins actually fluctuates strongly in time. So to a large extent, this heterogeneity between the different cells is due to intrinsic stochastic fluctuations of the underlying processes, in this case gene expression. But there are many other systems in nature that can be described as such stochastic processes. Examples include ecology, yeah, prey-predator models, epidemiology, modeling the disease spreading through a population, neuroscience, modeling spiking neurons, and so on. But how do we mathematically describe such processes? Let's start with the case where we ignore space. So we're not interested in where, say, molecules in the cell are, but we only care about how many of the different species are in the cells. And suppose we have a reaction system with certain species that undergo such transitions, or I call them chemical reactions. If we then ignore space, then the system is described by a Markov jump process, where the state of the system is simply given by a non-negative integer-valued vector, where each element simply counts the number of certain species in the system. And under certain conditions, it can be shown that the probability to be in a certain state X at a certain time is governed by the chemical master equation, which is such a differential difference equation. Okay, what about if we don't want to exclude space, but if we want to know where the particles are? Then one way to model this are stochastic reaction fusion processes, which I define in the following way. So I consider point particles performing diffusion or Brownian motion in continuous space and interact with each other stochastically. So they can be born, they can decay, they can interact with each other, and such bimolecular reactions can happen at a certain rate whenever the two participants are within a certain reaction range of each other. And also other reactions happen stochastically. Now, even though they are very simply defined, they give rise to very complex behavior and very useful, but they don't come without their problems and are very difficult to analyze. So first of all, typically there is no analytic treatment possible. Yeah? But and while it's very straightforward to simulate them stochastically, that becomes computationally quickly very expensive, because you have to simulate each and every particle in space performing Brownian motion and interacting with each other. And in particular for the problem of inference from experimental data. So suppose we have measurements of a spatial system and we don't know the underlying system and want to learn a model that describes the data, there are basically no inference methods available. And this last point is exactly the first part of my talk. So how can we do inference for such spatial stochastic models? And let me next first make clear what kind of inference problem I'm talking about. And let's assume that we have a cell with a nucleus. The gene produces RNA. The RNA becomes translated into protein, and the protein and RNA can decay. And now suppose we have measurements of the system, spatial measurements at discrete time points. So at discrete time points we get spatial points in space. In the beginning we might have only some RNA in the nucleus, and later here in red we might also see some protein. And now suppose we know that those reactions are happening, but we don't know how fast they are happening. So what we would typically want to do is we would like to infer the reaction rate constants. We might also not know the diffusion rate constants, so we might want to infer the diffusion constants. Or we might not know the exact reactions, and there might be potential feedback loops or something, and we want to discriminate between different hypotheses. Okay, how would we generally do this? The object that we in principle would like to compute is the likelihood. So let me define the likelihood next. So suppose we have some data at discrete time points t0 till tn, and let's theta be the model, which includes all the species, all the interactions, all the parameters, and so on. The likelihood is then simply the probability to measure this data given this model. Now in the most simple case, if we have no measurement noise and we observe all species, the likelihood simply factorizes into such a product of transition probabilities, yeah, because it's basically just a joint probability distribution. So, yeah, question? Oh.
It's just about your independence assumption. So these are measurements at discrete time points,
yeah.
And it looks like they only depend on the previous time point. Is that correct?
Yes, this is only true if the system is fully observed. So if you observe all species in the model, and if there's no measurement noise. Well, then it's a Markov process. Then the history doesn't matter. Only the state at this moment matters what happens in the future. If it's fully observed. If there's one species we don't observe or there's measurement noise, then this is not true. This independence.
I mean, even if you observe a system fully, wouldn't you get temporal correlations?
But for it's still defined as a Markov process. The propagation of the system in time only depends on the current state. The history doesn't matter, by definition. Thanks. Okay, so if we could compute this object, we could, for example, do parameter inference by simply optimizing the likelihood with respect to the parameters to find the parameters that best fit the data. But as I already said, typically there's no analytic treatment possible for such models, and simulations are very expensive. So suppose we would want to optimize the likelihood in a high-dimensional parameter space. Even one evaluation of the likelihood is very expensive because you would have to simulate somehow all those transition probabilities. So by just brute force simulation, that very quickly becomes infeasible. So we wondered if we can come up with an alternative method that does not rely on simulations. And I want to first give you the intuitive idea that we had in the beginning when we started this project. So for that, let's look at the data again. If we ignore what happens in between time points, so that those are molecules moving around, then what we get are spatial point patterns in space at discrete times. And there exists a class in statistics called spatio-temporal point processes that does inference for exactly such data. Of course, the points are correlated in some way. So, but since the data basically looks the same, we wondered, is there a connection between these two classes of models? Can we find a relation between our stochastic reaction fusion processes and point processes? And the answer is yes, of course, right? Otherwise I wouldn't tell you this. But in order to establish such a connection, we will need quite a few different ingredients, mathematical methods. So I first want to give you an overview of the path we have to go to establish such a connection. The starting point will be the chemical master equation, which we already saw, right? The stochastic description of a system when we ignore space. We will then use what is called a Poisson representation, which maps the discrete Markov jump process underlying the master equation on the continuous process. To then arrive at a spatial setting, we will use what is called direction diffusion master equation, which is basically a spatial discretization of a stochastic direction diffusion process. If we then take the continuum limit, we will get a single time description of our processes. And as it turns out, this will be related to Poisson and Cox processes, which are two particular popular types of spatio-temporal point processes. To do the inference for the type of data we want to, we will need multi-time points though, so we will next have to do a mean field approximation to arrive at multi-time distributions, which will then allow us to compute the likelihood very efficiently and make it very straightforward to do inference. Okay, we have already partially talked about the chemical reaction systems on the left side, and I think the audience might be less familiar with the point processes, so let's start next by talking about those. And let's look at the definition of a first spatial Poisson process. So suppose we have a region V, then a spatial Poisson process is a random process where each realization is a random point pattern in space. And the Poisson process does so with a certain statistics. Namely, if we take any subregion A of the volume, then the probability to find a certain number of points in this subregion is a Poisson random variable with a certain mean value. The mean value can be characterized by an intensity function, which is a non-negative real-valued function over the same volume V. And the mean value for any subregion A is then simply given by the integral over this intensity function. Okay, this is a spatial Poisson process. How do we derive a dynamical description? We let the intensity field evolve in time, for example, by defining it as the solution of a partial differential equation. And for each time point, we have a spatial Poisson process. Those models are very useful, but we will need an extension, something more general. So in order to do that, suppose that we don't have an intensity function that evolves deterministically in time, but that itself evolves stochastically in time. So for example, we could define the intensity field as the solution of a stochastic partial differential equation. And then for each realization of this stochastic field, we have a spatial Poisson process, which again is a stochastic process. So we kind of have a doubly stochastic process. And that's what's called a Cox process. And in that case, the probability to find a certain number of points in a subregion A is no longer given by Poisson distribution, but by a mixture of Poisson distributions, because the mean value is now itself a random variable, and it's still given as the integral over the intensity. Those processes are used quite a lot to do inference in many different systems, including epidemics, ecology, social sciences. But the crucial point here to make is that they're typically used empirically for systems that have no mechanistic underlying model, which in particular mean that the parameters of the model have no physical interpretation. They're just auxiliary variables that we fit to the data to then make predictions for the future. And that's why they seem to be quite unrelated to our stochastic direction diffusion processes, which are mechanistic description with physical parameters in the model. Okay, so much about the point processes. Let's now come to our chemical reaction networks. And let's start, just to quickly remind you, chemical master equation describes a non-spatial stochastic system by Markov jump process and describes the time evolution of the probability distribution. The Poisson representation makes the ansatz to write this solution of the chemical master equation as a mixture of Poisson distributions. And I give here for simplicity the expression only for one dimension, for one variable. And the mean value is a random variable which has a distribution. And as it turns out, this can always be done under certain constraints. And plugging this ansatz into the master equation shows that the Poisson variable u is itself a stochastic process governed by Largen equation or stochastic differential equation. And this is exact. There's no approximation. So in principle, one can, by doing this, map the discrete process underlying the master equation to a continuous process. However, one has to mention that in general, this variable becomes complex valued. And I don't know if you've noticed, we have seen two equations already, which looked very similar but in two different contexts. So we just said that the solution of the master equation can be written as a mixture of Poisson distributions in the Poisson representation, whereas the number of points in the subregion A for Cox process is given by a mixture of Poisson distributions. So there clearly seems to be some relation here. The problem is that the Poisson representation so far describes a non-spatial system, right? It's only counting molecules in the whole system. So we next have to lift it to a spatial model. And before we do that, we approximate certain reactions, because I already mentioned for certain systems the Poisson representation becomes complex valued and we want a real value description. And I don't want to go in detail, but just give you one quick example. So suppose we have a biomolecular reaction of this type, where this is the rate function. Then we replace this biomolecular reactions by two reactions, two decay processes for the individual molecules, but the rates scale with the mean value of the respective other species. So in some sense, it's a mean field approximation. And if we do this, then we arrive at a real valued Poisson representation. And this one we then want to apply to reaction diffusion master equation, which simply discretizes space into compartments and assumes that the dynamics in each compartment can be described by the chemical master equation, so by a non-spatial description. And the departments are coupled by diffusion, which are simply linear jumps in the master equations. Now, this one can always write as a master equation with a huge number of species by just relabeling, so one can always apply the Poisson representation. And that's what he did and studied the continuum limit. So let's look at a one-dimensional, one-species example. So suppose we have a one-dimensional interval discretized, and ui is the Poisson representation variable in compartment I. Then we already said that the time evolution of this variable fulfills a stochastic differential equation. We have here a discrete Laplace operator, which describes the jumping between compartments, a reaction part, and the noise term. And as it turns out, if one rescales the parameters appropriately and introduces this field, which is a function of continuous space, then one can take the continuum limit and arrives at a stochastic partial differential equation for this intensity field now, where we now have the Laplace operator in continuous space. Now this is not so exciting so far. The interesting part comes if we look at the probability distribution of molecules in a certain subregion of space. According to Poisson representation, this is given by a sum over the compartments of mixtures of Poisson distributions. And by using the property of Poisson distributions, one can show, after some manipulation, that it's possible to take the continuum limit, and then the number of points are given by a mixture of Poisson distributions, and the mean value is given by the integral over this intensity function u. And this was exactly the characterization of a Cox process. Oops. So for this single time probability distributions, we now already have arrived at our correspondence between the reaction diffusion processes and the Cox processes. And we don't just show that exists some Cox or Poisson process that approximates a single time distribution. We explicitly derive stochastic partial differential equations governing the intensity function. And I think this is quite remarkable because, as I said before, Cox processes are typically used empirically for systems where no mechanistic description exists. And we now have a description of our chemical reaction systems in terms of a Cox process where the parameters governing the dynamics of the intensity field are the actual physical parameters of the underlying system. Okay, for our original task of inference, this is not so useful yet because we need to get to multi-time distributions. And for this we need some sort of mean field approximation. And then we'll go in detail, but just give you the intuition. So let's look at those two classes of models. The reaction diffusion system, we have molecules that propagate in time and space. Whereas in the Cox process, we have random point patterns in space that are only correlated in time via the intensity function. So there's clearly a difference. Yeah, for the reason is the process representation only gives an auxiliary field for the intensity function. It's not the real state of the system. The molecules are the real state of the system. Whereas for Cox process, the intensity is the real state of the system. However, we still lift our representation to multi-time points, which again corresponds to, in some sense, the intensity function, because we give up correlation between time points conditioned on the mean field, on the intensity function. But if we do this, we arrive at a very useful result, because now we can use methods from statistics for Cox processes to the inference for our stochastic reaction fusion processes, which was not possible before. In particular, it's now rather straightforward to compute the likelihood, and we have an explicit expression. Okay, let's come back to the original motivating example. For simplicity, assume that we have only a one-dimensional cell and the nucleus is on one side. And I assume that we don't see the nucleus. So we have an additional free parameter, the size of the nucleus. We have the diffusion rates of the two species, and we have the reaction rate constants. And I assume that we have only a partially observed system, so we only measure the locations of the protein. And I simulate data, yes, so I simulate the process and then take 30 snapshots of the system, and then simply optimize, infer the parameters by optimizing the likelihood, which amounts here to just solving numerically a PDE. And to get some statistics, I do this 100 times. So I simulate 100 different data sets, do parameter inference of each of them independently, and then show here the mean value of those 100 inference procedures and the standard deviation in the parentheses. And the results are not super close, but considering that we have quite a large number of unknown parameters and only 30 measurement points, and only measure one of the species, I think the results are pretty good, with mean values quite close to the true value and reasonably small standard deviations. However, this system is... Yes?
So in a single realization, how good is your standard error? So on a single estimate, not on the 100 different ones.
Well, that's exactly the standard deviation, right, that we get from 100 samples. So this is exactly standard deviation for a single optimization. Yeah.
Okay, thank
you. Is it equivalent to consider 3000 time points instead of considering 100 realization of 30 time points?
Not really, because we start with here with no protein and no mRNA in the beginning, so we have a very strong transient behavior, but it converges to steady state. So if you would just take the same time interval and add many measurements, we would have a very long trajectory, but most points would be in steady state, which would not improve the inference very much, as one might think. So I haven't tested it, but I'm pretty sure it's more accurate to do it 100 times and then take the mean value. But it's a good question. I'm not entirely sure, actually. Now, this system is not so interesting yet, because there are no real interactions. Yeah? This is a linear system. So let's look at a more interesting example, an SIRS model, which describes the spreading of a disease through a population. So suppose we have a susceptible species that, up in contact with an infected species, becomes infected. The infected species can recover, and the recovered species can become susceptible again. And the setting we choose is we place lots of susceptible species randomly in space at time 0, point 1 infected at the corner, and then we see that over time the disease spreads through the population in such a wave. And first of all, we try to do parameter inference again, as for the previous system, that works pretty well. But then we wondered if we can go one step further, which was already one of the motivating problems I mentioned in the beginning. Namely, can we distinguish or differentiate between different potential models? So we assumed that there potentially could be an additional spontaneous infection, so the susceptible one can become infected without meeting infected. Yeah, question? Where's the microphone?
Okay, I don't understand what's the role of the last R2S.
Sorry?
R2S recovered to...
it can become susceptible again, so they don't stay immune forever, right?
Can't you combine I2R and R2S together? What would it change?
You have a delay.
Yeah, but if you change the RS parameter, it will be the same.
No, because you have a temporal delay. It's not equivalent in a stochastic setting.
No, if you change the small r and the small s rates.
Ah, those two. Well,
can't you combine it in one rate?
You mean go directly from I to S again?
Yeah.
No, it's not equivalent. You have a delay. If you look at the distribution of S, you get a delayed coming back. So I think you wouldn't get such a wave behavior, but I'm not entirely sure. But it's not equivalent. Okay, so we wanted to use our model, our approximation, to differentiate between those two different models. So those reactions, or the alternative model, those reactions plus a spontaneous infection. So what we did is we simulated data again, 100 time points, for both models. So one's having the spontaneous infection, one not having the spontaneous infection. Then we pretend we don't know which model data came from and use our model to choose which is the more likely model to underlie the data using Bayesian information criterion. And here you see the results on the left, where the true model to simulate the data did not have the spontaneous infection, and here where it did have the spontaneous infection. And everything to the right in the histogram means that our model picked the correct model. And as you can see, this is the case for more than 90%, and for more than 80% in both cases, we picked the correct model with a very high confidence, which is a pretty good example. And for this system, the computation time is quite interesting to look at. While a single realization of the reaction fusion process, so a single simulation of those points to simulate the data, took 250 seconds, the whole inference procedure using our method, so optimizing the likelihood here, in this case with respect to 4 or 5 parameters, took only 10 seconds. So our method is, in this case, highly efficient, and this also shows you how difficult it would be to use brute force simulations. If you, for one simulation, already need 250 seconds, you would have to run many simulations to even get one likelihood estimate. And this one you then still have to optimize on a 4-dimensional parameter space. Okay, let me summarize the first part. So as we've seen, there is a mathematical connection between stochastic reaction fusion processes and Cox processes. And as we have also seen, this can be used to perform inference. And the examples we looked at, the method turned out to be quite accurate and efficient. The next step we wanted to do is we wanted to use this representation not just to do inference, but to compute certain quantities of the process, certain path quantities. In particular, we were interested in first passage times. How long does it take for the number of proteins in a certain region of the cell to cross a certain threshold? And when we started looking at this problem, we realized this is actually very, very difficult. So we decided to first go one step back and study a non-spatial system. How can we approximate first passage times for a non-spatial system, described by the chemical master equation, to then later extend it again to a spatial setting. And this is exactly the topic of the second part. How can we compute first passage times for Markov jump processes? So systems where we ignore space and only count the number of molecules or different species in the system. Motivation, again, systems biology. For example, we could ask how long does it take for a cell to respond to a certain external signal. Yeah, we have an external signal, it binds to ligand. How long does it take for a certain key protein to become expressed above a certain level? Now, because we already said all those reactions happen stochastically, also the first passage time is a random variable. So optimally, we would not just want to know the mean value, but we would want to know the whole distribution of first passage times for the process to cross a certain threshold. But there are many other interesting examples. So to come back to our SIRS system, if we don't let the recovered species become susceptible again, the infected species will sooner or later die. And one might ask, how long does it take for the infected species to die? So in the beginning, here in green, the infected species might go up, but at some point it will die and become extinct. Now, the difficulty to compute first passage times is that it's a measure on the whole path. We want to know when the process crosses a threshold conditioned on that it has not crossed the threshold before. What it in particular means is that simply solving the free master equation, chemical master equation, is not sufficient unless we add some additional constraints, which make it harder though. And this in particular means that most of the approximation methods that have been developed for the chemical master equation cannot be applied to compute first passage times. And as before giving you the maths, I want to give you the intuitive idea that we had when we started looking at that. So suppose we have a process, say a number of proteins in a cell, and we want to know when does it cross the threshold 300. And now suppose we have an external observer that looks at this process and tells us at every time point if the process has crossed the threshold or not. So binary observation. You could think of it someone raising a flag, and as soon as the process crosses the threshold, takes the flag down. Now if I formulate like this is in principle a Bayesian inference problem. We have a measurement and want to know how likely is it to have such a measurement. And in particular, the probability to have a measurement that it has not crossed the threshold, which one could in physics for example call survival probability, is just 1 minus the cumulative first passage time distribution. Which means if we can solve this inference problem, we get the whole first passage time distribution. And this sometimes causes confusion, so I want to stress we don't need any simulations of the process and we don't have any real data. Yeah? We just pretend that we have measured that the process has not crossed this. For that I don't need any simulation or data, right? I just come up with this measurement, this binary measurement. Okay, let's introduce some notation. So let's x tau be the process and let's this be the whole path over a time interval. And let's call C the region of interest that we are interested in when we leave this region. And let's this denote an observation, the binary observation, that the whole path remained in this region C. Then the observation model is simply given by 1 when the whole path is in the region C, and 0 if it's not in C for the whole time. So simply such a binary observation. And the goal would then be to compute the survival probability. It's also often called partition function, which is simply marginalizing over all processes which we don't observe, right? We have an unobserved process. We only have this observation if it crossed or not. So we want to integrate over all processes so we get such a path integral. If we could solve this, then as I already mentioned, the first path time distribution is simply given by the negative derivative of this survival probability. Now this is not new yet, yeah? This is well known. The novelty comes by interpreting this as an observation process. So really treating this as an observer looking at the process and giving us 1 or 0. In that case, the survival probability becomes what's called the marginal likelihood in statistical inference terms. Okay, to compute this, we next discretize time. So suppose, the measurements, the time for the measurements. So suppose instead of measuring at all times, if the process has crossed the threshold or not, we do it only at discrete times. In that case, the whole observation process factorizes into single observations that are simply 1 if the process is below the threshold and 0 if the process is above the threshold. And if one of them is 0, then the whole observation becomes 0. The marginal likelihood can then be written as the probability to be below the threshold at a certain time point, given that one has been below the threshold for all previous time points. And those single terms can be computed in an iterative way in a Bayesian scheme as follows. So, because we have now only measurements at the discrete times, in between the measurement points, we have no constraint. We just have to solve the free chemical master equation. so which we just solve forward in time to get a predictive distribution of the process conditioned on the path measurements. Then, if we arrive at the next measurement point, we have to take this one into account, which we do by Bayesian measurement update, which gives us the posterior distribution at this time, taking all the previous time points and the current time point into account, which we get by multiplying the predictive distribution with the observation model and normalizing. And the normalization factors here are just the single terms that we had in the factorization of the likelihood. So if we solve this problem iteratively forward and keep track of the normalizations, we can compute the likelihood. And as already said, advantages, now we have to basically solve the unconstrained process between time points, so the free master equation, which in particular means we can now apply all sorts of approximations that have been developed for the master equation. And that's what we do. We use here what is called the normal moment closure to approximate the unconstrained process, which assumes that the multi-time distribution of the process can be written as a multivariate normal distribution with a mean vector and covariance matrix. And by using this ansatz in the master equation, one can derive ordinary differential equations for the mean vector and covariance matrix, which can be very efficiently solved in time numerically. For the update, we need a normal distribution again after the update. So what we do is we project this updated distribution again onto multivariate normal distribution by matching moments, by simply computing the mean and covariance of this object and then assuming again a normal distribution. That's what is called assumed density filtering in the machine learning community. Okay, this works quite well. Yeah, those approximations are very efficient, and we can now go forward in time. Yeah, question?
So I imagine that the normal approximation of the path will be even better when the time step is small.
For small time steps, it's better. Yeah, definitely. But that's a good point. Yeah, and in particular, this is an approximation if we do right to here look at the unconstrained process in between, because in between measurements we have some probability of the process going out and coming back. Yeah, so for large times this will be a very bad approximation. So in practice we would choose very short time steps. Yeah. But we started off with a continuous time system. Yeah, so we wondered, can we go one step further and take the continuum limit again to arrive at a continuous time description? And as it turns out, this is possible, but only if we do another approximation. Namely, if we approximate the hard measurement observation, the step function, by a soft potential of the form e to the power of minus delta t times some loss function or potential u, which is 0 below the threshold and then grows very quickly. For example, for the example before of crossing the threshold 300, we might choose the loss function to be e to the power of x minus 300. And the reason why we write it in this weird way with this delta t is because in the continuous time case, we would penalize the process by e to the power minus an integral over the potential over a certain time region. And this is just the discrete time version of this one at one time point. And if we do this, then it turns out after some derivation, a bit ugly derivation, we can take the continuum limit and arrive at a very nice compact result. We get ordinary differential equations for the mean, vector, and covariance function, and an additional ODE for the logarithm of the survival probability, or partition function. And they have a quite nice structure. The first term is the moment closure expressions that we would get if we would solve the free master equation without any constraints, without considering first passage times. And then we get extra terms that are derivatives of the expectation of the potential or the loss function. And those can be solved numerically, forward in time straightforwardly. And by in parallel solving this equation as well, we get the log partition function on a grid and can then just take the finite difference and get the whole first passage time distribution. Okay, let's look at an example, and let's look at the one we mentioned in the beginning already. how long does it take for species I to become extinct in such an SIR model? And here I show you the results for 4 different parameter sets. So different colors here are just different parameter sets. And the points are stochastic simulations, so simulating the exact system. And the continuous lines is our approximation. And we don't fit all of them perfectly, but overall I think we have a pretty good fit. And to be honest, I was surprised that we can even fit such a highly skewed distribution, and even here bimodal distribution pretty well. I did not expect it. And again, our approximation is very efficient in comparison to brute force simulations. So to compute one such whole first partial distribution, our method needs less than 1 second. Okay, let's look at another example, trimerization process. So we have monomers X, which bind to give a dimer, which bind again to a monomer to give a trimer. And we want to know how long does it take for a certain number of trimers to be generated, in this case 1000. So we can compute again for 1 parameter set the distribution, and it fits very well the simulations. And now, because our method is so efficient, we can very easily do parameter scans. And here we vary those 2 parameters and can look at the mean first pass time, coefficient of variation, and so on. And we see a quite interesting behavior here, almost a switch-like behavior of the probability to reach the target state. So in this case, one might end up with lots of dimers and not reach the specific number of trimers. Yeah? So the first partial distribution is not normalized in that case. So in some Fermat region, we get with almost 100% probability the desired number of trimers, and then we have quite a sharp transition to a region where we don't get it. And once more, we wondered if we can go one step further, namely if we can use this method to compute more general path properties. So for example...
Excuse me. Just a general question. How many realizations do you need to estimate the PDF or the first passage time?
You mean from stochastic simulations?
Yeah, many observations. I imagine that you're observing many different paths to infer the PDF.
I mean, for our approximation, we don't need observations. We just solve the ODEs once forward in time.
Yes, this is for the
exact results we simulate. That really depends on the system, how many species, how big the population are. Sometimes you need hundreds, sometimes you need thousands, 10,000. It completely depends on the parameter regime. It's hard to tell. Yeah. And it also becomes slower the more particles you have in the system, right? Whereas our method doesn't scale with the number of particles, only with the number of species, which makes it so efficient. Okay, but yeah, let's come back to the SIR system. For example, can we answer questions like, how likely does species I get extinct before half the population is infected? So putting an additional constraint on the process. So something like a conditional survival probability. Or how likely is it that protein A crosses a threshold before another protein B does? So those questions come up in statistical model checking, which they write as such an until operator. So how likely is it that property phi1 is true until phi2 is satisfied? So which means this, we have here a region in space of our species. We start in phi1 and want to know how likely is it that we end up in phi2 without ever going to the outside. So if we have 2 realizations, this one would not contribute because we leave the region before reaching phi2, and this one would contribute to the probability. And it turns out we can use our method to compute those, but only in discrete time so far. So unfortunately, so far we were not able to take the same continuum limit as before, which is a bit unsatisfying, but the discretized version also works pretty well. So here, let's look at a more complicated example. We have a genetic oscillator with 9 species, and we're asking the question, how likely is it that species 7, so the blue one here, stays below a certain threshold until species 9 here the yellow one, crosses another threshold. So how 1900 is here. How likely is it that the blue stays below until the yellow crosses the threshold 24,000? And this is a quite interesting example because it might happen in the first oscillation, but it might be that both don't reach the threshold. And then we get a contribution from the second oscillation, which is why the survival probability, so the probability to not leave the region at all, has such a step function. And we see that our approximation, the red one, approximates the exact result pretty well. And also for the actual until probability, it's not perfect, but we get a decent approximation. And once again, our method is highly efficient. So to compute this, our method took about 87 seconds, whereas the simulations take 120,000 seconds. And here we used in the computer science state-of-the-art software Prism to compute the until probability. Okay, let me quickly summarize. So we've seen that first passer chimes can, the problem of computing first passer chimes can be formulated as a Bayesian inference problem. And this allowed us to use some approximation methods to arrive at a closed set of ordinary differential equations for the first passer chime distribution and the process. And we were also able to extend it to more general logic constraints and properties. At the moment we're working on more general absorbing state formulation, which does not rely on discretization of time and which will hopefully allow us to derive more general and better approximations. But what I would really like to do is, what I started off with, right, is extend this to spatial systems, which unfortunately I haven't done yet, but that is the goal for the future. And let me finish by thanking my co-workers, Ramon Grima, Guido Sanguinetti, Boton Cecke and Dimitrios Milios, the ERC and BBSC for funding me. And finally, let me give you the references of this work, and let me thank you for your attention.
First, I want to congratulate you. It's fantastic.
Thanks.
So it allows me to like ask a provocative question. What if in the first part of your talk, what about this diffusion? You say that the spatial you have to solve this SRPD. So it's a diffusion equation. Mm-hmm. But what if it's not diffusive? Like, can you? Because this is an assumption, right? Like the fact that how the species they evolve in the volume or the space, mm-hmm. it's by diffusion. But in cells, for instance, you have other processes to make one species A to go from one point to another point. Mm-hmm. So it's not just diffusion; mm-hmm. There are motors or whatever, mm-hmm. And so. How could you like either extend, or maybe I didn't understand, and this is already in what you did, but if it's not just pure diffusion, like that there are other processes that
Yeah. No, you're completely correct. We have only pure diffusion processes so far. It's a very good question, and we wondered about to what extent we can extend the method. And for stuff like active transport, it would be quite difficult. If we have chemotaxis, for example, I think should be possible. Yeah, we would get drift diffusion equations and similar, but we haven't looked concretely into it, so it would be not so trivial to extend it. So as it is now, our method only really works for pure diffusion.
Also, even if it's not active, like also in the cytoplasm, it's not like a particle diffusing in a liquid, but it's so entangled. There are fibers everywhere. Mm. So
in a crowded environment, kind of.
Yeah. So I have seen work where they end up with it's a diffusing mechanism, but it's in a complex environment, so they become like fractals and stuff. So this may be less complex than having some. Well, it's just different kind of diffusion, but maybe closer to this, but still.
So it would be great if we could extend it to those settings, but I think it's very difficult because the way we derive the representation in terms of direction diffusion mass equation assumes that there are points. So giving them a spatial extension would be very hard to achieve. If they are in a crowded underlying environment, that might be possible. You know? Then we would just get some explicitly space-dependent terms in the SPDEs, I think. But to include volume exclusion of the particles that we model themselves, I think would be very hard. Yeah.
Thanks.
I would like to come back to earlier questions. The first one is, between your observations, you actually have to evolve the free master equation. And you actually said, well, there is a probability that you can go above threshold while this. And obviously you can make your time step very small to make that probability very small. But then I assume if your time step becomes very small, that just makes your numerical scheme more expensive. So I do wonder whether there's some sort of optimal sweet spot where essentially you can work with a decent delta t and still have your sort of error term that comes from the free evolution of your master equation to be negligible in a controlled fashion.
That's a very good question, and we wondered about how to find the optimal time step. The good thing is, in the first part, for first passage times, we don't need it, right? Because there we were able to compute a continuum limit, and which kind of combines the solution and the update. So in that case, we don't have to do this anymore. In the second part, we still have to do time steps, and there we actually have to compute numerical integrals for the until operators. So there, yeah, we would want to take a time step as large as possible, right, to make it numerically efficient. And we don't have a rigorous way yet to estimate this time step. But it's so efficient, the method, in comparison to simulations, that we basically just decrease the time step until we saw convergence. Okay. If it takes only 80 seconds, you just go smaller and it converges quite quickly. But still would be nice to either, I would really love to compute a continuous time version also of the until operator, or to find an optimal good estimate for the time step. Yeah, that would be great, but so far no idea how to do that.
Okay. And my second question relates to the normal approximations that you make. How well controlled are these approximations? And obviously you use some moment closure techniques, and there are various other approaches how you can close the master equation. So my question is this the optimal closure for the current application? And do you have any feel for how good that normal approximation works?
That's a very good question. It's the optimal method, yes and no. Yes, in the sense that's one of the few that works, so that where we could derive the equations. I think no, because it's actually quite inaccurate, I think, for first passage time distributions. Because what you do is, in the process, you always cut off your process, and even if the unconstrained process would be well approximated by a normal distribution, if you cut off a Gaussian and approximate again by a Gaussian, that doesn't seem like the best approximation to make, which is why I was so surprised that it actually performs so well. So I'm sure there's room for improvement by using other closure schemes that approximate a constrained distribution better, but the ones I looked into so far did not allow me to take this continuum limit again, because we use a lot the properties of a Gaussian. So that's also one thing I would want to do in the future, to improve the approximation by this more general formulation that I mentioned in the end, where we don't have to discretize time. And then maybe we can use more general closure schemes to arrive at a similar but better approximation. Regarding the intuition, when the normal moment closure does well, for unconstrained systems, yes, that's quite easy, right? The more nonlinear system is, the more particles you have, the better it becomes. Unfortunately, for this constrained process, I have no intuition. So I thought I had it in the beginning, but then it completely failed when I looked at the experiments. Because sometimes it did better for very nonlinear, very small number systems. So unfortunately, I have no intuition. So hopefully, with the more general formulation, we would also be able to get more intuitive results, or maybe, hopefully, optimally even error estimates. But that's really future stuff.
Great, thank you very much.
I guess if not, let's thank all the speakers for this morning.